some background of biological elements in the context of the cell. So we start a little bit on the level of biology, because I think it's important that you all get on the same scene and that you get an idea, basically, of the material that you are working on. Yesterday we heard already how beautiful red blood cells are, that you can use red blood cells, for instance, for very experiments that give you insight into the mechanics of the membrane, that gives you insight into the shape changes of membranes. And today I would like to take you more into the basically eukaryotic cell that contains also a lot of intracellular organelles. Red blood cells are an exception of eukaryotic cells because they have lost all their intracellular organelles. They have basically removed the nucleus and are only left behind with a plasma membrane and the cytosol. But all other cells in our body, they are nucleated cells containing a lot of intracellular organelles. So let's have a look on a typical eukaryotic cell. What you see here, it's an animal cell, just a little bit illustrated. And what you see is basically that inside this cell is separated in different compartments. Maybe you even remember from the school time, one big compartment would be the nucleus that is surrounded by a nuclear membrane. But there are many more compartments. For instance, the mitochondria that actually contain a double membrane system. So it's an organelle that contains two membranes. But you also see there's the Golgi apparatus, there's membranes that are basically forming peroxisomes, there are membranes that are forming lysosomes. And all these different membranes basically allow this compartmentalization, allow the generation of organelles, and all these organelles have different tasks. So on one hand, that is beautiful, because that allows us to run the metabolism of the cell in specialized parts, but it creates also a problem. Because once in a while you need to exchange material between these organelles. And that means the membranes need to become permeable for a particular substance. And we will later discuss in this course which basically elements are responsible to allow the selective permeability of the membrane. So for the organelle, just as a reminder, if you take the cytosol, there the synthesis of proteins takes place, in particular of cytosolic proteins. Not necessarily of membrane proteins, that's a very minor part, but the major part of protein synthesis in the cytosol is basically for the cytosolic proteins. Then the nucleus, you know, it's the place where the DNA is placed. It's also the place where the ribosomal RNA is produced, an important component later for the formation of ribosomes. And it's also the place where the DNA is synthesized. So the mitochondria are considered as a power station. So they produce actually the ATP of a cell, and for that they have a sophisticated machinery of proteins embedded in the membrane, an aspect that we will later discuss when we go on the lecture for bioenergetics. If you take the endoplasmatic reticulum, that's the place where all lipids are synthesized. Let's say almost all. Whenever I say all, I over-saturate it a little bit, but most lipids are synthesized at the endoplasmatic reticulum. That's also the birthplace of membrane proteins. It's interesting, no? So the majority of membrane proteins, it's born in the endoplasmatic reticulum. But that poses a problem. How would then the protein get, for instance, to the plasma membrane, when it would be a plasma membrane protein? How would this protein get to the lysosome, when it would be a lysosomal protein? So you see that cells obviously need to find solutions for this problem, that they have, let's say, centralized the synthesis of lipids and centralized the synthesis of proteins, of membrane proteins at the endoplasmatic reticulum, that they need to find a solution how to build up the other membranes. So almost all lipids that are synthesized in the ER, then some of them need to be used for the synthesis, for the growth of mitochondria, for the generation of lysosomes. So there's a lot of processes required, exchange of material required between the different organelles, because there's a clear separation of tasks. So if you take the Golgi apparatus, that's a type of sorting station, where basically the cells decides should the material be delivered to the lysosome, to the plasma membrane, should it actually stay in the Golgi, or should it be basically sent back even to the endoplasmatic reticulum. So lysosomes, it's a place where things are degraded. So sometimes cells also not only need to synthesize, but they also need to degrade material
And that's a particular, basically, organelle that is equipped with enzymes that can do this job. And peroxisomes are also a type of very special organelle. They are very responsible for the beta oxidation of fatty acids, for the breakdown of fatty acids. So now when you look at this, that means, okay, we have basically a complex system of intracellular membranes, and the first question you can ask is, what would be the lipid composition of all these membranes? Is it the same? Is it different? Or you are, for instance, interested in particular processes in one of the organelles. So how can we isolate these organelles? So let's start basically with the question, how can we study the various lipid compositions? So how can we study organelles, just in very general point? So one thing what you can do is basically a process where you take cells or tissues and you break them. That would basically release the organelles, and now you can take advantage that these organelles differ in their weight. Some membranes contain a lot of proteins, they are very heavy. Other proteins contain more lipids and very few proteins, so they are very light. So that means you can basically break cells, then you have a cellular homologate where all the organelles are free, and you can basically run now different centrifugation, which we call differential centrifugation. So you use different centrifugal forces to segment your organelles. And when you do this, you get basically pellets at different speeds that differ in their organelle composition. So that gives you already a first idea that basically of separation for the membranes. Okay? Now you see there's also one problem associated with this. The problem is to open the cells, because when you are too harsh, you even might open up your organelles. So it's not easy to isolate intact organelles. But if you are only interested in the membranes themselves, you might not care. You say, okay, I just destroy even or open at least the organelles, but I still have the organellar membranes and as basically sheets in the preparation, and I can isolate them by this centrifugation. So typically this centrifugation is not yet good enough really to have a pure organelle preparation. So the next step what you would do is you can basically use a so-called density gradient. So what you have here is, for instance, different sugar concentrations, and that's then, you can say, sometimes you see this also with cocktails. If you go in the bar, then they make these different layers of colors. The very sugar-rich part of the cocktail is on the bottom, then the lighter part is on the top, and that looks very beautiful because they use also different colors like strawberry and whatever, apple, then you get nice color. You can do the same here with basically a type of sugar gradient, and you put your organelles on, and then you start to centrifuge. And now the organelles will move to the area where they have then the same density. So that's basically density centrifugation, and an outcome you can see here. That's, for instance, a preparation from yeast cells where you have separated here the different organelles, and you can see that they basically end up in these type of bands. And those you can collect, so you can selectively collect this band, that band, and there, depending on the density, you, for instance, get the mitochondria, or you get the peroxisomes, or you get the plasma membrane. And also, depending from the organism, there might be differences. In case of yeast cells, the plasma membrane is very heavy. In case of human cells, the plasma membrane is very light. So there's also species-dependent differences. But when you have basically this knowledge, you can basically collect in these bands. You can, of course, also check, for instance, for so-called marker proteins. So some proteins are localized only in particular organelles. And then you can take advantage as you have antibodies, for instance, against these proteins that you can check. Would this band here, for instance, correspond now to the endoplasmatic reticulum? And would this band here, for instance, correspond to mitochondria? You can imagine that mitochondria are very dense, two membranes, and very heavy, so they would basically go very down to the bottom. The endoplasmatic reticulum membranes are very light. They would rather stay more on the top. So now this allows you to basically have the different organelles separated. Also here, again, there is a problem. This separation is not complete. You always have contaminations. So whenever you basically look in the literature about lipid compositions, you should be also aware that, of course, the technique by which basically these organates have been isolated will determine also the end result. So when you see a result like the lipid composition of the mitochondria is as such, it doesn't mean necessarily that this is the absolute truth, because it could be that in this preparation there are other contaminations from other membranes. So that's something you have to be always also critical to the data in the literature, because there's a lot of steps. No? You need to open the cells. You need to isolate the organates, and none of these steps is perfect. So the end result that you get will also not be perfect, because the limitation is here the quality of your separation. So another technique that you can use is so-called free flow electrophoresis. So that's a process where you actually take advantage of the charge differences between organates. And that has something to do, for instance, with the lipid composition, also with the protein composition. And depending whether these organates are strongly charged, less charged, if you put them in an electric field, they will start to move in this electric field. Those that are very charged would be, very within this one time period, move very fast. The others would be left behind. And that machine here is basically a chamber. You can see here, that's the chamber. So it's a chamber that has two electrodes on which you can put basically an electric field, or actually voltage, to generate an electric field. And then you have here inlets. So, and you have basically here a type of buffer flow. And what you can do is basically you have this buffer flow that's here just indicated with these colorful fluids, where we have basically added just some color in some of those channels. And this is basically constantly running, and you could basically now put a voltage on the two electrodes to generate an electric field. And when you do this like that. You would basically have here, for instance, your injection point of your organelle, purified organelle preparation, and you let it enter the chamber, and basically this preparation will float into the chamber. And once the preparation is in the chamber, you switch on the electric field, and now the different organates will basically run in this electric field depending on their charge. Highly charged, very fast; less charged, less fast; non-charged will actually not run at all. No? And then you can here in the end collect what comes out. So you get a separation based on an electric field, and you know actually this technique also maybe as a type from the time in the bachelor, like SDS page, where you use a agarose gel, or in this case an SDS gel, to separate proteins. Now in this case, basically you don't use any detergents. So you want to basically not disrupt the membranes or anything. This is a detergent-free method, and you can recover your sample afterwards. That's why it's called free flow electrophoresis. You can basically recover your organates and work further with them. You don't destroy any function of the membranes, besides that you basically have isolated now the membranes. Okay, another trick that you can do, now it becomes a little bit biological, you can take advantage of basically particular mutations. That's very basically common, for instance, in baker's yeast, which is also a type of model organism for biologists, where you basically introduce mutations in proteins that are important for the vesicular transport, for the communication between the organates. And these mutations are basically temperature-sensitive mutations. So you mutate amino acid that is, for instance, involved in forming an ion bridge in a protein. And that is important for the stability of the protein. If you remove this ion bridge, this protein might still fold, but it becomes now less stable. And when you raise the temperature, the protein might unfold, so it will lose the function. And that's basically here one example. You have here a protein, it doesn't matter the name, it's a protein that is important for the docking and fusion of vesicles. And this protein is functional, carries a mutation that makes it still, or keeps it functional at low temperature. But when you move these cells to a higher temperature, the function is actually lost of this protein. So it will not do. So that means you can grow your cells under low temperature conditions, and in the moment where you move them to a higher temperature, the function is lost, and now all the vesicles start to accumulate. And these vesicles are vesicles that are basically destinated to the fusion of the plasma with the plasma membrane, but they can no longer fuse. So now the cells will fill up with a lot of vesicles that are all would like to fuse with the plasma membrane, but they cannot do this. So what you isolate now is secretory vesicles, but compared to wild-type cells, you have these vesicles extremely enriched in these mutants. And you basically can come up with other mutants. There's also mutants where basically the transport from the endoplasmatic reticulum to the Golgi is blocked so that you can accumulate another type of vesicles. There's also basically mutants in mammalian cells that you have the same type of phenotype where you can block particular steps in the transport pathways, which allows you to enrich, let's say, intermediate compartments that are used for the cell, for instance, to transport material. So also in this way, you can basically now, when you open up those cells, isolate these vesicles. And the opening up, we have not yet really discussed. No, there could be this harsh method, like a hammer, or we use also glass balls. It's like if I would throw stones on you, and then you would basically open up. But if you do it too harsh, again, then the problem is you also destroy eventually the organates. So there's always a compromise between soft opening and keeping still the organates intact, but harvesting a lot of material. Yeah. Osmotic shock is another example that's often used. No? So I don't know whether, for instance, when you are bleeding, we have learned yesterday, no, in our blood there's a lot of red blood cells. And when you are bleeding and you wash your hands, then typically it looks like everything is red. And as if you have lost liters of blood. But in fact, all the erythrocytes, the red blood cells, they just burst in the water. And then the hemoglobin comes free. And hemoglobin is so much red. You know, it's such an intense color because of the heme that is associated with the hemoglobin, that even in high dilution it looks still very reddish. Yeah? So that's why and lysis is a very soft method actually to open up cells and to allow organates to come free. Okay, so now depending on what membrane you are interested, also what type of organism you want to study, you can use different type of techniques to isolate membranes, to enrich for membranes, and that gives you the material that you can work with. You can use maybe such organates already for the opti trap that we have seen yesterday. No? Jabari has introduced us, for instance, in this wonderful microscopic technique where you can trap vesicles, where you can trap cells. In this case, you could, for instance, think to trap these secretory vesicles if you want to study them. In this trap, you can also test, for instance, mechanical properties. So actually with this technique, you would basically now here be able to isolate some material that you could start to study. Now, what is the next step? If you are interested in lipids, you would like to isolate the lipids. That's a technique probably you all are familiar with because you know that lipids have a strange property. They are quite hydrophobic. They like water also, but they're also quite hydrophobic. And this you can take as an advantage because lipids like organic solvents, while all the soluble material likes typically the water phase. No? So what you can do is you can basically now take your preparations, either the whole cell or the isolated membrane, and you can basically extract the lipids by chloroform, a mixture of chloroform, and we do this typically in a two-step phase. So you take a mixture of chloroform, methanol, water that is basically forming one phase. So chloroform can be mixed with water as long as you have a little bit methanol with it in the right proportion, and that gives you here such an extract. And then you add an excess of chloroform, and that leads to a split of phases. So you get type of organic phase, the chloroform phase, and you get here the methanolic water phase. And now, depending on the properties of your basically material, the material will either end up here, or depending if it likes both, you get basically a distribution. So the lipids you would typically find here, okay? But that's also not true for all lipids. No? So some lipids, when they have very hydrophilic head groups, then you get a partition. then you get basically also, you might lose some lipids in the water phase also. Yeah? So that's why this, it's a very common technique for most lipids, but some special lipids you would avoid to have this phase splitting. You would rather like to work then only with a one-phase extraction. Okay, so now when you have the lipid extract, you have basically removed all the proteins. And just to make sure, why you have removed here actually all the proteins? No? One may wonder, what happens to the proteins here, Thomas? So the point is, when you basically add chloroform methanol, it's a very hydrophobic environment. And under those conditions, the proteins will unfold, they will denaturate. So what will happen is basically, if you basically treat salts with chloroform methanol, the lipids become free, and the protein, the methanol removes the water from the protein, and that makes them unstable. They start to unfold, and then they start to aggregate. So you get protein denaturation. And that will lead to the fact that actually the proteins will be here as a layer. Between the two phases will swim on this layer as an aggregated pellet, so to say. And that you leave behind, because in this case we are not interested in the moment for the proteins, we are only interested for the lipids. If you are interested in the proteins, oh, you could take those, but they are typically too aggregated to really work further with this. Protein extraction you would do in a softer way if you are basically using, for instance, SDS page analysis and that. Okay, now we have the lipids, and then you can do thin layer chromatography. Maybe you have done this even in school. You can do for all substances that are basically multi-components, you can separate on that way, or you use basically lipid chromatography. And then in our times now, these basically pre-separated extracts, they go to mass spectrometry. And then you can determine actually exactly lipid composition. You know? The amount of lipids is a little bit difficult in this technique, mass spec, but you can still do this with, you add standards where you have a defined amount of basically lipids added, and then you can determine also not only what type of lipid, but also the amount of lipid in this extract. Okay, that you could do now for all the organelles I have shown you. Yeah? Okay, and let's do this maybe for two organelles today. So if you do this, you end up here, for instance, for the endoplasmatic reticulum, with a membrane that contains hundreds of different lipids. And these lipids are classically phospholipids, glycerophospholipids. We introduced them yesterday. But you see also there are some steroids. So in case of an animal cell, that would be cholesterol. No? In the case of a fungal cell, it would be ergosterol. In the case of a plant cell, it would be primarily cytosterol. No? And you see basically that here on the color, okay, it's at least 5-6 different major type of lipids. So phosphatidylserine, phosphatidylcholine, sphingomyelin, in this case not yet, because we only have here the glycerophospholipids. Okay? If you do the same for the plasma membrane, then you see, oh, this lipid composition looks different. There's a new type of lipids, which is absent here. And those are the sphingolipids. And these are here glycosphingolipids. They are decorated with sugars. So this membrane is completely different in the lipid composition compared to here. But I told you that all the lipids are synthesized here. And what you also see is basically the steroids. Here are steroids, but also here. But here's far more steroids. What happened here? So how can it be, when all the steroids are synthesized here, that this membrane is highly enriched in steroids, but here is hardly any steroid? So there must be sorting going on, right? The cell basically produces lipids in this organelle, but then these lipids are not equally distributed to all the other organelles. So there must be particular sorting steps, and we can discuss basically what type of sortings and later. So the first important thing is the lipid composition differs between the different organelles. Here in the extreme case, as an example, the endoplasmatic reticulum, the plasma membrane. The endoplasmatic reticulum has very low levels of sterol and doesn't have higher glycosphingolipids or higher sphingolipids. They are absent. plasma membrane, a full contrast. It's very rich in steroids and has a lot of glycolipids. I don't know whether you can already guess. I mean, you know, in nature is always a reason why this is like. Maybe you have already a guess why this is basically the reason. Do you remember what the particular property of steroids would be? If you add steroids to a membrane, what properties would change? Fluidity would change, right? And if you have normally a very fluid membranes with a lot of lipids with a lot of unsaturated fatty acids, and you add steroids, what would happen to this membrane?
Liquid order.
Yeah, it would actually become more condensed. No? We even don't talk yet about domains or so, but it would become more condensed. No? And now you can ask, okay, does it make sense to make this membrane tight and this membrane not? And for the plasma membrane, you would consider it makes fully sense, right? You want to have the cell really separated from the outside. So this membrane should be tight, very tight, should be protective. This membrane, here the endoplasmatic reticulum, when you would need to make all the membrane proteins there, that means you need later, and we will talk about this, you need to insert proteins in this membrane. Or you make lipids. These newly lipids need to be integrated. Then this membrane needs to be very flexible, right? It should be actually not stiff. It should allow that something can be inserted. So this is the biological reason for these different lipid compositions. They give these membranes different properties, but that's very important for their biological function. Only due to this fact, they can serve these different biological functions, okay? And now when we take this to synthetic biology, you can again take advantage of this. No? By knowing this, you can create your own membranes with your own properties, where you say, I want to have a property of very flexible, that I can insert something, or I want to have a property of very stable, because I want to include something which should never leave this vesicle again. No? So this, basically, this knowledge from biology will help you also to design model membranes for particular purposes. No, we have heard that some of you are interested in drug delivery, so this is basically something that will help you to create such systems. Okay, so now basically there's one more thing. So we have only talked about the lipid composition in the moment, but we have not talked about the lipid arrangement. Yesterday we introduced this term lipid asymmetry, which is already here illustrated. No? There's a particular set of lipids only on the outer leaflet that is absent. But how has this been discovered? Or how would you analyze this? No? Also, there's a couple of techniques, and the very classical technique is basically based on the chemical modification of lipids when you have a substance that cannot penetrate. So if you, for instance, add to the cell outside a substance that can modify lipids, but not penetrate, it will only modify the lipids on the outside, but not on the inside. And if you compare basically treatment, non-treatment, you can determine basically what type of lipids have reacted with my substance and which lipids have not reacted. And one example is here this trinitrobenzosulfuric acid. That's a substance that reacts with amino groups. It's just as an example. So this substance can very difficult penetrate membrane. It stays outside. But it reacts with free amino groups. So some lipids have free amino groups. If you remember from yesterday, there were, for instance, a lipid phosphatidate serine containing a serine which contains an amino group, or phosphatidate ethanolamine, free amino group. So if you add basically the substance and these lipids would be present on the surface, it would basically covalently bind, react with these amino groups, and you get a modified lipid, a new lipid, due to this reaction. And then you make thin layer chromatography. That's here now a two-dimensional chromatography where you have separated the lipids with one solvent in one direction and a second run with another solvent in another direction. That gives you quite a resolution of different lipids. Here again shown for glycerophospholipids, different glycerophospholipids like phosphatidate serine, phosphatidic acid, phosphatidate inositol, phosphatidate choline, phosphatidate ethanolamine. And now you do the same analysis with membranes or cells that you have treated here with TNBS. And when the organelles are intact, the cell is intact, TNBS can only react to the outside, but not enter to the inside. And you see then you get basically here a new lipid coming up, which is a modified lipid due to this reaction with the TNBS. And the amount of this relatively to the total amount of the unmodified lipid gives you an estimate of how much was exposed to the outside versus how much was protected to the inside. And when you do this analysis, then you come up that basically the aminophospholipid, and this is phosphatidase serine, phosphatidase etanolamine, that they are basically localized on the inner leaflet and not on the outer leaflet under normal conditions. Now, we have heard that this can change. There can be a collapse in lipid asymmetry, but that would be typically a type of abnormal situation, or it would be a situation that basically body wants to use to recognize particular cells, for example, old red blood cells, where you also would get this exposure of aminophospholipid after about 100, 120 days. So a red blood cell in our circular system lives about 120 days. Then it needs to be removed. No? Just from the biologic point of view, why does it only live 120 days? These cells can have lost mitochondria. These cells have lost endoplasmatic reticulum. These cells have lost the nucleus, no DNA. So they cannot produce any protein anymore. They cannot make any lipid anymore. The only thing what they can do is basically, with all the hemoglobin, they can transport the oxygen. Well, they can also transport carbon dioxide, but not much more they can do, because they don't have anymore the machinery to renew proteins and to renew lipids. And that leads to the fact that, of course, when they basically in our blood are circulating, that lipids get damaged, oxidized, protein gets damaged, and then at a certain moment, these cells would actually completely disintegrate, but before this happens, they are typically recognized by macrophages and removed. That's the reason why they only live 120 days. Okay.
So you said 120 days or only under 20 days?
120. 120 days. Yeah. Quite long, no? Yeah. Well, relatively to that, they cannot really renew the membranes. Yeah. Okay, so now basically I want to take you a little bit in the lipid dynamics. Okay, so let's go back to the point that we have here the endoplasmatic reticulum, that here all lipids are synthesized. But the material for the lipid synthesis is found in the cytosol. Okay, so if you want to make a glycerophospholipids, you can play this evening or this night if you want. You take the units. You take a glycerol, you take 2 fatty acids, you take, for instance, phosphoric acid, and you take, for instance, serine. And now you want to build this all together. The serine is an amino acid you will find in the cytosol. Phosphoric acid, it's basically also in the cytosol. The glycerol is in the cytosol. From the glycolysis, you can basically produce it. The fatty acids are made in the cytosol. So everything is in the cytosol. So that means the lipid synthesis, when you want to make new lipids, it will happen here on the cytosolic leaflet of the endoplasmatic reticulum. So you only build one monolayer. So you would basically make here lipids, 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 lipids. You would expand one layer. That doesn't give you a membrane. Because with a membrane, at least half of the newly synthesized lipids need to be moved to the other side, right? But I told you this hardly happens, because there was this problem, flip and flop. It's very restricted. These lipids do not like to move the head group to the hydrophobic core. That's energetically very, very unfavorable. So there's the first problem. How to make a membrane when all lipids are only made in this layer? And the solution is that you need a protein. You need a catalyzer that is able to basically move these newly synthesized lipids to the other side. And those proteins are basically very unspecific. They allow many, many lipids to exchange. And we call them scramblases or all-term endoplasmatic reticulum flipases. But in fact, they don't work with energy, they just work like a mixer. So now, lipids that are produced here can move to the other side, and that allows a stable growth of the membrane. But at the same time, this would mean this membrane here, it's a symmetric membrane in terms of lipid distribution, right? If I mix any newly synthesized lipid that can move to the other side, in the end, if I do this here with all of you, today you are sitting like that, tomorrow you are sitting in another way, and again, so there would be, no asymmetry. But this membrane is asymmetric. So how do you solve now this problem? How to make from this membrane an asymmetric membrane? Okay, so basically the point is that you use active transporters, flipases and flopases, that now move specific lipids, only to the inside or only to the outside. And you avoid that those proteins ever make it to the plasma membrane, right? So these flipases here, or scramblases, they should be either not in the plasma membrane or they should be inactive in the plasma membrane, right? And if this is the case, then those transporters would start to sort. They would say, you are a lipid that needs to be there. You are a lipid, you need to be there. You are a lipid, you need to be there. So now I basically place you on two different sides of the membrane and I get an asymmetric membrane. And this is what cells do. Yeah? Okay, there still is one point I have not discussed with you, right? Thomas, but what happened here to these glycolipids? They suddenly appear. And the reason is that they are synthesized in the Golgi apparatus. So they are introduced later. So you basically get the lipids produced, you make a lot of endoplasmatic reticulum, then you can basically bud vesicles that move these lipids to the next organelle, and the next organelle can grow. But there you introduce now a new type of lipid, these sphingolipids. And that allows basically to generate a new lipid composition, and these flip and flopases allow to generate an asymmetric distribution. Okay? So there is a reason why the Golgi exists. It's a filter station. It basically is a sorting station that separates the plasma membrane from the endoplasmatic reticulum. So this is the place where membrane proteins are synthesized, where the lipids are synthesized. Most of the lipids are synthesized. Membranes are then transported to the Golgi, and here new lipids are synthesized, glycolipids are synthesized. And in this place now, you get basically the flipases, flopases starting to work to allow an asymmetric membrane to be created. So you have basically here a membrane that the role is protection and communication. And you have here a membrane, the role is synthesis. And this membrane, it's basically the Golgi system. It's communicating. It's like a diplomat. This should stay in the endoplasmatic reticulum. This should go on to the plasma membrane. So there is a reason why this organelle appeared. That helps basically the whole cell to keep these different lipid compositions in the membrane. Yeah? Okay, so now you get an impression basically there are different lipid compositions, that there's a reason for that. It has strict basically consequences for the functions. And if I take you shortly to bacteria, how would it in bacteria? I don't have a slide, but we can just discuss this. What type of bacteria do you know? Do you want to give me a name or just some general? Do you know a bacterium? Did we hear something yesterday? We heard something about phages, bacteriophages that are actually Escherichia coli, E. coli. Yeah, that's a very interesting bacterium because it's already quite complicated. It has two membranes. It has a plasma membrane and an outer membrane. Now, all the people that are studying microbiology, they would of course say, Thomas, it has an inner membrane and it has an outer membrane. That's true. But the inner membrane, it's the plasma membrane. That's the plasma membrane. That's the only membranes, right? Do you also know other bacteria that are less complicated? Yeah, so what would this type of bacterium? Yeah? Is it gram-positive, gram-negative? Do you remember maybe a little of those? Yeah, and what would that mean? Gram-positive, what would this mean? It only has one membrane. It only has a plasma membrane. Huh? So that would be the very, very simplest bacterium. Only one plasma membrane. Okay, so where are these lipids then made in the bacterium? You want to say? Or you want? Yeah, of course the precursors, so the fatty acids are made in the cytosol, the glycerol is made but the lipids, they are not water soluble, right? If you make a lipid in the cytosol, you would have a problem. That will not work. So they need to be made on the plasma membrane, right? It's the only membrane left. So they are made on the inner side of the plasma membrane in the bacterium. But now again the same problem. On the inner leaflet you make all the lipids. How do you make the outer leaflet? So you need a lipid transporter. Then the outer leaflet can also grow. Okay, where would you make the membrane proteins in such a bacterium, in a gram-positive bacterium? Because also a bacterium needs channels, needs to exchange material, also in the plasma membrane. So what would be the property of the plasma membrane if you want to be now a designer of nature? What properties would you give to the plasma membrane of a bacterium? Should it be flexible or should it be stiff? You want to give a keyword? Or? Flexible, no? Because you want to insert membrane proteins. You want to insert lipids. But what do we wish from the plasma membrane? Rigid. Should be protective. Oh, what a dilemma. There's a real dilemma for the bacterium. It's the membrane which basically everything should happen. It's a very metabolically active membrane, but it should be also very protective. So you see that there is a limit for bacteria, also from the point of complexity, what can happen on the plasma membrane. Because the plasma membrane, if you need to have very flexible, but the bacterium, actually the best would be a very protective plasma membrane. So what was the solution for the bacterium? With the gram-positive, they make a very thick cell wall. So that's the protection. So bacteria have a very, very strong cell wall because they cannot make the plasma membrane basically protective, because that's the very metabolic active place. So what happened in the eukaryotic world was now the separation of these tasks. So what normally the plasma membrane of the bacterium does now here do the endoplasmatic reticulum. And the plasma membrane can be now very protective, and we don't need a cell wall. And that opens up completely new things, because now cells can start to communicate directly. There's not basically this layer of cell wall in between. You see? Plants have, of course, another problem to solve, right? What is the problem of plants? Plant cell. They need stability. They need to grow. I mean, some trees I have seen also here in India, so some trees, wow, you know, how stable are they? It's not by the plasma membrane. No? I mean, even the plasma membrane of a eukaryotic cells, yesterday we saw the erythrocytes, the red blood cells, how flexible that can do these undulations and so, which is good in our case, because if you want to move red blood cells to these small capillars, yesterday we heard some capillars are only 3 millimeters or maybe 1 millimeter. So the cells actually, they need to be able to deform that they really fit through these nice capillars, that they can nicely flow through this. But for a plant, the plants need a cell wall to give this stability. And they have another trick. They have a strong vacuole that creates a pressure that presses the plasma membrane against the cell wall, and that allows a strong stability. So there's different solutions in nature for basically having membranes that are stable or more flexible, and whether you need to place them to the outside or not. So bacteria, they were limited in their capability simply due to the point that they put all the tasks on one membrane. And in the eukaryotic cell, you put different tasks to different membranes, and that allows basically far more complex chemical reactions to occur. Okay, so now maybe one last point that I would like to discuss, it's lipid dynamics. So how can we actually monitor the movement of lipids between membranes or across membranes? And one trick we are using is, for instance, that we modify these lipids. You see here a fluorescent lipid. It's a modified lipid that carries here a fluorescent group. Okay? There were also other examples yesterday from Tripta's lab where you can add, for instance, also a fluorophore here on the top. So there's, depending on what precise mechanism you want to study, you can use lipids that are modified with fluorophores at different places of the lipid molecule. So if you want to study, for instance, the flip-flop, you don't disturb here the head group. But you can modify here this type of fatty acids with a fatty acid that carries a fluorophore. And in this case here, the fatty acid is a very short one. So you have a lipid with a long fatty acid and a short fatty acid plus a fluorophore. So this lipid, what would you think about the hydrophobicity? Would this be basically very hydrophobic or less hydrophobic compared to a lipid that would carry 2 long fatty acids? Yeah? You want to guess? Yeah? Less? Yes, would be less hydrophobic, because the shorter you make the change, the less interaction. No? If you have a very long fatty acid chain, and basically the other lipid also has a very long fatty acid chain, they can interact by van der Waals forces, and also in terms of water molecules, they don't want to interact with water molecules, so the forces that brings them together are very strong. No? If you make one chain shorter, then they start to become more water soluble. No? But in this case, it's nice because they like still membranes, but they are partially water soluble, so you can basically resuspend them in a buffer and add them to cells, and then they will insert in the membrane, and that's here mammalian cells under the microscope, fluorescent microscope. Yesterday we heard about the fluorescent microscope, where you excite basically your fluorophore, and then you look for the fluorescence that is emitted, and you see that these cells basically are here completely now labeled. And when you incubate these cells longer, then something happens. Then these lipids, fluorescent lipids, suddenly appear in the cell. And they can appear in this pattern, but they can also appear in this pattern. So it depends from the type of lipid you are using. Lipids that are basically transported by these flipases, they will show this type of distribution. As soon as you basically move the lipid by a flipase to the inner leaflet, these lipids can redistribute because they are partially water soluble to the other organelles, and you see completely labeling of the inside of the cell. Lipids that are not transported, they can only be taken up by a process that we call endocytosis. So it's a process where the lipids are basically trapped in vesicles, endocytic vesicles, and you only see here these little dots, these endocytic vesicles. So with this, you can actually monitor whether a lipid can easily cross, flip-flop, no, in this case flip, or whether it can only be taken up by endocytosis, for instance, by fluorescent microscopy. There's also other techniques when you want to be more quantitative, techniques like flow cytometry, where you basically can easily analyze also the quantity, the amount of fluorescence that is associated with the cell. Okay, another example is here shown for basically yeast cells. These are yeast cells. This is phase contrast. No? Yesterday you have heard basically microscopy, one technique is phase contrast, and then you can look under the fluorescent mode which of those cells are fluorescent, and in this case this lipid, when you add it to yeast cells, is also flipped and accumulates here in the mitochondria. So that's basically here an accumulation, staining of the mitochondria. If you take, for instance, another fluorophore, a lipid-like fluorophore, that is basically not flip-flopped, it stays with the membrane, and it can only enter by endocytosis. And here we have incubated the cells under conditions that the endocytosis is blocked, so all the fluorescence, all the material stays on the plasma membrane, nothing is transported inside of the cell. And with these type of fluorescent molecules, you can, for instance, then now see to which organates they are transported, where do they enrich, are they anyway at all basically flip-flopped, or they are only taken up by endocytosis. You can do this with a type of other here modifications. These are basically spin-labeled molecules, spin-labeled lipids that you can detect with a technique of EPR, or electron paramagnetic resonance technique, that goes quite for physics, but some of you are physicists. So you take basically advantage of an unpaired electron that has basically in a magnetic field a momentum that you can detect, right? So not going into more detail, but of course if you wish we can do this also in the tutorial or later in the evening to go more on this technique. So that allows you also to basically follow this lipid. And when you take this, for instance, here for red blood cells, no? I think it's a very nice example because it's only the plasma membrane. And you have these type of lipids, then phosphatidylethanolamine and phosphatidylserine, they are rapidly transported to the inner leaflet, while the other lipids stay outside. So this is basically here an example for a transporter, for a membrane transporter that selectively moves particular lipids to the inner leaflet, and by that keeps lipid asymmetry. So this transport process, it's well functional in young red blood cells, but this mechanism will stop in old red blood cells, and as a consequence, the lipids, phosphatidylserine, are not anymore transported back, stay exposed. Macrophages can eat, for instance, those cells. This process is also used for immune cells, for instance, to basically signal with each other. So you can consider this basically like you have an ion gradient across membranes. In this case, you have a lipid gradient, particular lipids outside, particular lipids inside, that can be collapsed. Yeah, just for those that are a little bit interested in lipid transporters, because this is a little bit my particular field, many of them are actually in diseases involved. You have here diseases on the fatty acid degradation, adrenoleukodystrophy. You have basically problems in eyes, degradations of eyes when these transporters are not working. You have liver problems, bile formation problems, because all of these transporters are responsible to maintain basically the proper lipid asymmetry in membranes. And if this is not working proper, it has consequences depending in which tissue it basically takes place for the whole functioning of the tissue. Yeah, so in the last minute, I would basically just remind you on this fluid mosaic model. We yesterday heard about this, and this is what typically you would think about a membrane, right? So classically, you have the bilayer. In this bilayer, you have basically lipids that in the plane can move. They can form also little domains. Also on the inner leaflet, this can happen. And you have membrane proteins embedded, and that can basically move and swim. So typically, the example is that you say, if you compare this with the ocean, the lipids, the ocean are the lipids, and the icebergs or the ice mountains, these are the proteins. And that would be our idea of a membrane. I want to challenge you a little bit with this. It's a good model, and we can use it, but is this really, it's a model. So do membranes really look like that? If you consider, for instance, okay, if this is true, then that would mean all membrane proteins would have exactly the same part here, the same size for passing the membrane. What was it? About 4 nanometers. But what about membrane proteins that would only be 3 nanometer? Or what about membrane proteins that have a hydrophobic part that would be 8 nanometer? How do you fit them in a membrane? Of course, this is not the question that I come up with. That has been driven already other researchers. And there came basically a model that is called the matras model. I don't know whether you have it here in Indian, but in my bed now, the basically matras, it's very flat. But sometimes you can find matras that are, because they are basically, you know, held together with little stops, that it goes like little waves. And that's indicated here. So that would be basically going on like that. So you have basically a lipid mixture of very different lipids. Lipids with long fatty acids, tails, short tails, and that would allow membrane proteins to associate with those that have short tails. Is this maybe a better model? Is this an addition to the fluid model? Not everything is like in the ocean that ice mountains are just swimming and the ocean would be flat. Is the ocean maybe really stormy? And you have icebergs very big and very small. Is this not better picture? But even then, is this picture then true? Are there so many lipids? So if you all would be proteins, there would be enough space between you, no? But what about membranes where, you know, let's say we would have the 10-fold more participants, but we would only have this space. Then they would start to sit there, to sit there, to sit there. Where would be the lipids then? That would be a membrane almost only composed of proteins, right? So in fact, typically this model, it's a little bit too simplified. Membranes are actually very packed. They are very, very packed, and they are basically not at all homogeneous, like this would basically imply. And then, of course, comes the next. You can have these demixing areas where you get basically particular proteins, particular lipids associated. But this is eventually also not stable, but dynamic. So it's maybe like now in the tea break that we will soon have. Some of you will cluster, others will cluster. And then maybe after 5-10 minutes, some of you will leave to another cluster or will actually stay around without not clustering at all. And the same happens in the membrane. But the problem is to actually analyze this, to detect this, because some of those domains are very short-living. And it's very difficult to detect processes that are very short-living. No? If I would have, for instance, a camera and I would make every 5 minutes a photo of the tea break, then I would see Tamil there, Tanmai there, Tanmai there. But that would be difficult then from these 5 minutes to really, what was your trajectory? What was your pathway? But if I have a technique where I make, every millisecond, click, 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 I can have a movie. I can watch exactly them. But how to make a movie every chink and all those elements make it very difficult to analyze those processes. But it's super important. So I don't want to basically demotivate, but I rather would like to motivate to go for these questions and try to analyze it, because it has really a tremendous basically impact, this understanding, also for signaling processes, to cell communication, all aspects that we will discuss later in this course. Yeah, so then I come to the summary. So if you want, biological membranes are asymmetric, but you already learned not all membranes are asymmetric. Some are more asymmetric, plasma membranes, some are less asymmetric, endoplasmatic reticulum. You can consider them as a solution. That's maybe something we can also discuss later with Abhishek. No? If you want to simplify, yesterday you have seen that sometimes it's helpful just to make a simpler model of a membrane, just to make to get a basic understanding. And we have these two basically layers of lipids, a bimolecular lipid layer. Many, if not most, physical properties can be explained simply by the different lipid compositions. And there's important aspect, this type of shape of lipids. And we have heard about a parameter of the shape, no? This form parameter P, that you can use basically to determine what type of lipid would like to make a micelle, inverted micelle, or a membrane. Then lipids and proteins are quite mobile. I haven't shown you this, but you would basically believe me in the terms of, since they are not covalently bound, just non-covalently, you have these possibilities of movement. And the basically lipid asymmetry requires the concerted action of flipases, flopases, and scramblases. And lipid molecules are not evenly distributed, but basically also are able to form microdomains. Okay, so with this, I would like to thank you very much for the attention, and we can use the tea break for more discussion.
So just a quick comment. So the models that Thomas talked about, we will also show some data.
Very good.
So there is a tea break now.
Yeah, and feel free in the tea break. You can come to me. No, it's not. You don't need to hesitate to give me any free minute here. So, Tripta is taking care for the evening that I have some free minutes.
Also, this attendance sheet that you are signing, I'm circulating it again when you come back. Just check your spelling because I'm going to write exactly this name in the certificate and the grade sheet. Just check once again the spelling. If it is wrong, then just make a note. Okay, done. So we can do that after we come back.
One, two, three. Okay, welcome back. So now we make a big jump before we discuss biological membranes, and we will do more of this in the next session, but this session is now actually designated to lipid vesicles. So we want to start basically a little bit to look into artificial membranes that are forming the basis for instance, studies to understand the complexity of biological membranes on a simpler model system. And when you look, there's many different type of model membrane systems that you can consider. I just have basically listed here 3. You have these type of planar lipid membranes that you can build. You can build nanodiscs, an aspect that we will basically at the end of this course discuss. And you have basically liposomes. And today now we will shortly start to focus on these liposomes, an aspect that then you will also later in this session in the afternoon have a practical work on. And yeah, if you look basically on those systems, there's one clear difference from vesicles to all other model membrane systems, and this is they actually define a compartment. So the vesicles have an inside and an outside. That's very important, for example, when you are interested in transport processes. If you want to know how a substance transported across membranes, then this system, it's an ideal system. It's also, it's very similar to the biological situation, where basically you also have in cells type of moments where membranes are forming vesicles. Okay, so let's take such a vesicle, and now let's categorize them. So at the first you can basically group vesicles, for instance, according to their size. That would be here these small unilaminar vesicles, large unilaminar vesicles, and giant unilaminar vesicles. And unilaminar means you only have one bilayer. That would be the situation for a biological membrane. And you see they differ quite in size. The small ones, it's so up to 7 nanometer. 70 nanometer is something you cannot see anymore in a light microscope. Yesterday we have heard, no? 200 nanometer, it's basically so the border where you can see with a light microscope. So here you would need electron microscopy to make them visible. No? Large unilaminar vesicles, still very small, typically too small also for a microscope. The only trick what you can do is you can add a fluorescent molecule, for instance, to this, and then you would see light, but you would not see the dimension. You would only see whether the vesicle is there or not. But then when you come to giant vesicles, then we come here to the level of a cell. 100 micrometer is basically a very large cell already. Red blood cell was 6 micrometer, typical mammalian cell 20 micrometer, 100 micrometers already very, very large. So those you can basically watch under the microscope, and you have seen yesterday some nice examples from Tripta's lab where you can basically, under the light microscopy, follow vesicles and analyze them. And some of these photos you might have seen, basically, they also contain some different type of vesicles, vesicles that contain vesicles in themselves, so-called multivesicular vesicles. Maybe you have also looked, sometimes you can recognize multilaminar vesicles. Yeah? So this would be basically a situation that you would typically not find under biological conditions. And if you look at this, is that not something wonderful? Is this not almost a model of a eukaryotic cell? Would this not be vesicles in a cell? So you can imagine, if you think about synthetic biology, that would be a type of first start to an artificial cell with a compartment. So you can use the whole space of different type of vesicles depending on the application, depending on your research interest, and to select whether you would to prefer with the small ones and or with the large ones, whether unilaminar vesicles would be the right choice or whether multivesicular vesicles would be the right choice. Now practically, how do you make such vesicles? Well, if you would go now on the lab, in the afternoon you will go on the lab and you will get also some examples to how to make them. So it typically starts all that you select lipids that you can typically buy, or you have good chemists that synthesize these lipids. And then you can basically add them in a glass tube. Typically they are chloroform soluble, and you will need to basically evaporate just the chloroform. And when you do this in such called evaporator, rotation evaporator, you get a tiny lipid film, a very thin film of lipids around the glass. And that's good, because this very thin lipid film on the glass, when you now add buffer, you can basically shed off vesicles from this lipid film. So these lipids will basically dissolve and form vesicles, but from yesterday, you remember, we would need to use a particular type of lipid. Not every lipid would do this. What type of lipids would we need to use to get these type of vesicles? Cylindrically, huh? Or you use the right mixture. You mixture cone shape, inverted cone shapes in a right ratio, and then you would still get vesicles. So if you use this procedure and you want to have vesicles, there are some restrictions. No? Not any lipid forms this. If you use this procedure with cone-shaped lipids or inverted cone shapes, you get inverted micelles or micelles or hexagonal structures, as we have discussed yesterday. And Abhishek also explained what are the reasons and what are the constraints, just based on the geometry. So now when you basically want to force these vesicles to be unilaminar, because here this would be basically a mixture of vesicles, just by adding them and shaking, you would get shedding off of vesicles that are unilaminar, multilaminar, multivesicular, different sizes, large, small, whatever. But now when you want to have a very defined size, you can use, for instance, sonification. So sonification means you use ultrasound. Ultrasound in the solution, and that will basically lead to the fact that you shortly disrupt membranes and they reform and you get unilaminar vesicles, but very small. So you put so much energy in that at a certain moment the structures that you have generated are left with an unilaminar membrane as a very small vesicle, typically not more than 50, 70 nanometer large. But it's very easy to make in the lab, because often in labs you find these sonificators also for cell breaking or for getting, for instance, substance in solutions. What you also can do is now, you can basically use this lipid film that you have hydrated with your buffer, and you have formed shedding off of membranes, you can basically use this vesicle suspension and put it to a small filter, a filter with very small pores, 100 nanometer, 200 nanometer, depending what type of vesicle size you would like to do. And when you basically force these multilaminar vesicles through these very small pores, then a miracle happens. Suddenly, after this filtering, you get vesicles of around 200, 100 nanometer size, unilaminar. That would be large unilaminar vesicles. And this process of filtering, it's basically here a type of metal holder in which you place filters and where you press your lipid solution through. This process we call extrusion. So you extrude your lipid suspension, and as a result, you get LOVs. We will also do this in the course so that you get an expression how to do this, and you can also build this together so that you just get a feeling. So just for now, just to remind yourself, okay, there's a process that we call extrusion that can generate us large unilaminar vesicles. So now you would have vesicles, and you think everything is fine, and you can start to work. Often, actually, the problem can start because depending of the lipid compositions of these vesicles that you have generated, they might like to stay in suspension, or they might like to start to aggregate. So what do you think if you would, for instance, make lipids, also vesicles with lipids that would be charged? So let's say you take a negatively charged lipid, and you generate your vesicles with this lipid. Would these lipids like to stay in the suspension? There's one person who says no, Thomas. Other ideas? Let's say you would be negatively charged, and I would be negatively charged. Would we like to come together? What happens when we both are in a solution with a lot of calcium chloride? Still? Do you want to be the calcium? Okay. How many charges do you have as a calcium? +2. Okay. Do you want to come up? Do you want to come up for the stage shortly? Only if you want. Yes, you come to me, you are the calcium, and you are the other lipid, negatively charged. Yes. Okay. You are floating here around. I'm basically a vesicle, having a negatively charge here. Huh? You are a vesicle, where's your negative charge? Show your negative charge. Yes. So, and what would happen when we come together? Whoa! Yes. 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 You are the calcium. Where are your positive charge? Ah, okay. So what do you think will happen now? Oh, now we are together. Now we start to aggregate, right? So a solution without calcium, we will be stable. The vesicles will stay stable, right? Negatively charged, negatively charged. Now you put us into a solution with a positive ion, multivalent, magnesium, calcium. Where's our calcium? Whoops, now we aggregate. So, you have to be just thoughtful. This could be interesting because maybe you want that the vesicles aggregate, but if you don't want this is a disaster, right? This would be a very problematic situation. Thanks a lot. And the same happens if you, for instance, have vesicles with sterols and non-charged lipids. They can be the surface can be basically, because it's uncharged, there's the danger that these vesicles will start to adhere and stay together, to aggregate. And that sometimes even depends also from the temperature. And this is the example here, where we basically stored vesicles at 4 degrees at room temperature. No? Let's say this evening you will make vesicles. Then you are very happy, you put them in the refrigerator, you go home. And then you come back in the next morning, and then you see this. And then you run to Thomas, Thomas, look. And I say, what a disaster. All your vesicles are aggregated. what would you do? Any idea? Would you be in tears, or would you start again, or what would you do? Sonicated. So you could sonicate. What would be the result when we sonicate? Remember, we had large unilaminar vesicles that we extruded. Now the suggestion was sonication. We could do this, and what would be the result? We would get small vesicles. We would still have vesicles, but completely different. The size would be now different. So that would work, but you don't have any more large unilaminar vesicles. You only have small unilaminar vesicles left. Could be still interesting, depending on what you are working. Could be a disaster, and Thomas would say, oh, you know, we discussed, we need here large unilaminar vesicles. We cannot work with this. You could think about resuspension, no? Pipetting up and down, pipetting up and down. But if they are really aggregated, that will not help. So you would need to start again. I would say, sorry, you need to, the whole work from yesterday, you need to go ahead. Yeah? Or you have found out not to store them at 4 degrees, but rather leave them at room temperature, where this process has not happened. So that's something, when you make vesicles, just be aware, it's not that they are basically, you know, completely stable. It depends, again, from the lipid type, the lipid mixture. It's not that they don't interact. Also, that depends from the buffer composition. So that's all, let's say, depending what you would like to investigate. We, in our lab, for instance, we would like to investigate ATP-driven transporters, a group of proteins we will discuss later. They need ATP and magnesium. So magnesium, no, divalent cation, might trigger aggregation. So then you have to be very careful with the lipid composition and to think. So sometimes you might like to include something. That could be an indicator, or it could be a drug. So how do you do this? No? So far we have only discussed how to make a small vesicle, how to make a large vesicle. Now, in the first glance, it's actually relatively easy. What you do is you add the substance that you would like to include into your buffer. So you add this buffer to your lipid film. You start with the lipid film, lipids, you know, from the chloroform solution, evaporated, nice film on the glass. And the reason is why you would like to have a film is that you have a large surface of a very thin layer of lipids that can interact with the buffer, with the water molecules. And you would have your dye in, and then you do a process that we call freeze-thawing. Freeze-thaw, freeze-thaw. And that puts a lot of stress on your vesicles. They will eventually rupture, close, or you make them a little bit more permeable, and then again tight, permeable, tight. And that will allow the dye and the buffer to enter into exchange. Of course, there's a limit also of the materials, of the size. No? So typically, dye molecules are very small molecules, the indicators. But if you want to include a protein, that might be too harsh. No? If you freeze-thaw a protein, it might denaturate. No? You know already with an egg, you know, the boiling and so, it's also very sensitive, or egg and acid. If you take an egg and you put a lot of acid in, it also partially denaturates. So proteins are sensitive, and that would be too harsh. But if you have a small chemical molecule, this process works very well. So you would have basically typically something like dry ice or liquid nitrogen. So dry ice minus 70 would also work. And then you can do a water bath, and then you change always between basically the dry ice and the water bath, or the liquid nitrogen and the water bath. And then you basically, in the end, extrude. What was the secret of the extrusion? You want to make similar vesicles of similar size. So you want to remove these multivesicular vesicles, and you want to remove all basically different sizes and unify them. And now you have your dye outside and inside. That could be sometimes enough. But typically, you would like to remove now everything what is outside. And for that, you use a process that we are calling gel filtration. So it's a process where you have a little gel that is pores. And this gel has different sizes of pores. Pores that are very large, pores that are very small. And then you add now your vesicle buffer solution on top, the vesicles are very large. They will go very fast to the large pores. The dye will go to all the pores, and it will lag behind. So very fast, basically, your vesicles come out, and your dye is basically left behind. You can then rerun the vesicles to make really sure that they are free of dye from the outside. And then you have filled vesicles. Vesicles that are basically only contain your indicator. And now you can start to use these vesicles. One example is shown here, where we used a dye called calcein. So this calcein has here basically in the molecule carboxyl groups that are basically oriented in the way that they, for instance, can bind divalent cations, for instance, kappa. And that will change the fluorescent property. So this calcein is a fluorescent probe, but the fluorescence will depend whether it has bound an ion or not. Yeah? So the fluorescent will be very strong without an ion, but when you add kappa, the fluorescent properties will go down. And if you, for instance, now add kappa to the outside, you see that this kappa, it's actually slowly going in. So it's not completely impermeable, the membrane. Yeah? But if you add, for instance, a kappa ionophore, you see then you can, it's really fast permeable because the fluorescent drops down, and when the fluorescence of this calcein drops down, it's an indication that kappa has entered and has bound to this dye and changed the fluorescent property. So why could this be interesting? You know, the group that we have worked with, they were interested in these ionophores. Because you know that antibiotic resistance, it's a serious problem, and that we are actually running out more and more of antibiotics against bacteria. But bacteria are, for instance, for kappa, are very sensitive. So if you have, for instance, now a particular ionophore that would allow ions to pass, then you would have another way of extra treatment. So there's a lot of chemists that try to synthesize different types of ionophores as antibiotics, as new antibiotics that would make bacterial membranes permeable. And imagine you would create an ionophore that only makes bacterial membranes permeable, but not mammalian cells. Then you have a fantastic antibiotic because you would only kill the basically bacteria. So the challenge then for the chemist is, of course, to think about ionophores. What properties would I need to add to this ionophore that it only targets the bacteria, but not the mammalian cells? No? But for instance, you then, if you are more interested in biophysics, you would be interested, does the ionophore at all allow ions to pass, or which ions would it allow to pass? Because you could also use now other type of ions as long as they can bind to the calcein and change the fluorescent properties. No? We were actually quite amazed how permeable membranes can be also for ions, because the classical view is that ions do not cross at all, and for many ions that is true, but for some ions, depending also on the type of lipid, this is now a liposome with a very simple lipid composition. It's not a biological membrane, it's only one type of lipid. And that's actually, it is resistant in a way, but compared for the ionic condition, but it has permeability to kappa. Slow, but it is there. Okay, so now the other aspect that we have not yet discussed is the giant vesicles. How do you make the vesicles so large and unilaminar that you can see them under the microscope, that they would be 10 micrometer large? Now there's several methods for that. You will learn here today one method, and that is based on electroformation. And that's a technique I will explain you in a minute. Maybe let's first look, is there any advantages of these giant vesicles versus the small vesicles? Yeah, first of all, they are more easily to analyze under the microscope. And that allows you basically, for instance, to study membrane properties like domains. So you can make these giant vesicles not only from one lipid type, but from mixtures of lipids. And we learned as soon as you start to add 3 lipids, in particular when they belong to different classes, to the sphingolipids, to the glycerolipids, and to the steroids, then there's the possibility of domain formation, depending on the ratio of mixture. And you see this here. That would be, for instance, a vesicle where our lipid marker is equally distributed. That is a vesicle where the lipid marker is localized only in one part of the vesicle, indicating there was a demixing. And this lipid marker has now mixed with one part of the lipids, but not with the others. You can study lipid-protein interaction. That's from the biological point very interesting. I also have heard yesterday that some of you are interested of peptides, how peptides are interacting. So what you can basically here study with, it's basically whether a protein binds, whether it penetrates, or whether it actually makes these vesicles leaky, destroys these vesicles. We have used this often for proteins that interact with lipids. So we have proteins that only can bind to a particular class of lipids. So you make vesicles with this lipid and control vesicles without, and then you can test, does this protein really bind to this particular lipid or not? And sometimes it's surprising, proteins that have been claimed to be super specific, at least in these assays, suddenly you find that they are not only recognizing one type of lipid, but often also 2 or 3 types of lipids. Then you can start in this system very nicely also these membrane deformations and budding. So some proteins interact in the way that they help to deform membranes or that they help to actually budding off membranes. And that's something you can here study under the microscope under well-defined conditions, right? Or you can basically here also study transport mechanisms when you include membrane proteins, an aspect that we will discuss later, how to get actually membrane proteins in such vesicles. No? So all these aspects here, you can basically study just on the vesicles made of lipids. Other aspects like those, you need to make GOVs containing then these membrane proteins. Yeah. Maybe also just as a reminder, you can also use this system here for protein-lipid interactions. If you are interested, for instance, whether a protein makes leakage or whether a protein induces fusion of vesicles with each other. In this case, you would, for instance, see this calcein load in very high concentrations. And when fluorophores come together in very high concentrations, then there is a process happening that we are calling quenching. So not all the energy that you put into the system will be emitted as light. And that's in particular in these high concentrated solutions. So if this vesicle now then becomes leaky and the calcein leaks out, this quenching will stop and you get a huge increase in fluorescence. So in this case, an increase in fluorescence will be indicative for a leakage of this dye. And that you could basically, when you have peptides, you want to know whether this peptide will make this vesicles leaky, will penetrate. You could use this assay also on small vesicles and analyze this. But you can, of course, do this also on the giant vesicles. The reason why I went back to the small vesicles is, it looks like the small vesicles are very easy to make, right? You only need lipids, you add buffer, maybe you need to sonicate or you need an extruder, but in principle they are very easy. The giant vesicles, they are a little bit more difficult to make, and they are also a little bit less stable because they are very large. So you have to think also about your question. Not, I don't want to say GOVs are now the solution for everything, because it's quite hard to work with them, and you always need to define, is this question better to be studied on giant vesicles, or maybe LOVs or SOVs are fine enough to be used. Okay, so here's one example from our lab where we make basically vesicles of different lipid composition. This is a vesicle only containing phosphatidylcholine. This is a vesicle containing phosphatidylcholine plus phosphatidylethanolamine. That's a vesicle also containing a little bit phosphatidylserine. And for those of you that are not so familiar with these chemical operations, the other 2 letters indicate the fatty acid composition. So this is a D-oleoyl phosphatidylcholine that tells you to have basically 2 fatty acids based on oleic acid. Okay, so this is only for those that also when you read papers that you get a little bit familiar. It's nothing I would say you need to do for the examen, but just that you get a feeling of all these different names. Okay, so now we add basically a probe here. It's annexine. It's a protein that is normally intracellular localized. And this basically said that should be specifically interact only with phosphatidylserine. No? And what you see is basically this protein we have fluorescently labeled. So the protein, it's in high dilution here, poly to C, but when it basically binds to the vesicles, it binds to the whole surface of the vesicle, and then it's basically, you can see it as a nice fluorescent ring. And you see that this only happens here for the vesicles with phosphatidylserine. So that would be an indication that this protein really doesn't bind to phosphatidylcholine, phosphatidylethanolamine, but only to the lipid phosphatidylserine. Yeah? So that would be a typically protein-lipid interaction assay you can do under the microscope in this case. What you also can do is this domain formation. No? Remember, if you take, for instance, sphingolipids, steroids, and glycerolipids, these sphingolipids and steroids, they will form an extra phase, a liquid ordered phase, while the basically phospholipids, the unsaturated glycerophospholipids, stay in a type of lipid disordered. And these are the two phases. So if you now add a fluorophore, which loves this area, it will accumulate here. And then you basically see it accumulates here in this nice staining. So that is a very stable domain formation. In the real cell, such domain formations are very, very rare in this extreme. Because this is a long-living domain. You can see after days still under the microscope. In biology, I told you these domains are not that stable. They are actually, formed small. Then they disappear, come again, disappear. Only some organelles, for instance, the yeast vacuolar membrane, that is considered as a very type of raft-like membrane, where you have a very stable domain formation. But all biological membranes, all others are typically not in this extreme case. But still, this is a nice model system because you can ask questions like, does my protein bind more to this type of environment or more of this if you add, for instance, a protein that would interact with lipids, right? Okay, so how do we make them? I taught you use a technique electroformation, and you will be introduced in Tripta's lab this afternoon, how this in reality looks like. So you can take these type of plates here, and you will get introduced into these plates. They have special properties. They can conduct basically electricity. No? And then you can add basically your lipids on it. You form a lipid film. And when you have basically made the thin lipid film and you form a chamber and you add a voltage, an oscillating electric field between the two glass plates, then a miracle will happen. You get this shedding off of vesicles, of very large vesicles that look like that. Some will be unilaminar, some will be multilaminar. It depends also the type of lipids that you are using, the type of buffer, the type of condition, whether you do it in the heat, whether you do it in the cold. So you can modulate this a little bit. Another technique is basically you take here these wires, metal wires, on which you deposit, for instance, your lipids, your lipid suspension. Then you get this thin lipid film, and you put on these wires again an electric field, so a voltage that generates an electric field, and you get this shedding off of vesicles and end up with this in the microscope. And if you do well today, you will see this. You will do this by yourself. So you will be able after that to come to my lab and do GOVs. Or you stay with Tripta and you can make a lot of GOVs in the lab. And that could be then interesting for membrane investigations, protein-lipid interactions, whatever you like. Yeah, I think with this, I would end here, and I don't want to give far more secrets because the real secrets come in the lab where you get a hand-on training so that you get a more practical feeling also how to do this. Okay, any questions? I think we still have a bit time. Not yet. Okay, do you feel like I'm not sure whether I take too much of it. I just want to make maybe a little thinking with you. Not on model membranes, but just back. If you want, you can stop also recording. That's up to you. How do we do this here? Can you help me only to tune down the beamer? And then we use basically the whiteboard here. Okay, do we have to clean this? Yes, thanks a lot. And then we just shut down here. Yes. Okay, so let's go back shortly for the cell. We had this eukaryotic cell. You remember there was this animal cell where we had basically here the endoplasmatic reticulum. And where we had here the plasma membrane. Okay, if this would be a plant cell, what would I have needed to add to my drawing? Cell wall, yeah. And which organelles would we find in the plant cell? You are too fast for me. Yeah, what would you suggest? Vacuole, yes. What would be in the animal then the equivalent? No, the lysosome. No? And so in our cells it's the lysosome, and we have basically in the plant cell the vacuole. The vacuole is basically a type of equivalent, type of waste place, but it also has additional function like this turgor pressure. So vacuole. Then I have heard chloroplast. Ah, a complete new organelle. That's actually a very fascinating organelle. No? It has 3 membranes. We will talk about this because it's important for the generation of energy, how plants actually use the energy of light to produce ATP. No? So that's actually the major difference. Chloroplast we would need to add, 1 more organelle, and the cell wall we would need to add. No? Very important. Okay, so now I told you that basically most lipids are basically synthesized here in the ER. And this would happen only on the cytosolic leaflet. So here you would basically add fatty acids, head group, fatty acid, head group, and this layer would grow. But we would need to move half of the newly synthesized at least here over, and that would be by the help of scramblases. That would be unspecific. So now lipids are on that side, lipids are on that side, quite symmetric. And now we want to make a plasma membrane where we have basically, besides of lipids only here, and lipids, other type of lipids on that side. And we learned that this is basically with the help of flipases and flopases done. No? But we also learned that here is very little steroid synthesis. So the synthesis of steroids is here. Do we have another color eventually? Let me see. Yes? Is this good enough? No, better take a yellow maybe. But here you have a lot. Most of you have neighbors. Can you discuss for yourself a few minutes? How do you have ideas? How you can basically generate from a membrane that has a very low steroid content, a membrane that has a super high steroid content. Also, all the synthesis of this happens here. A It's a bit contradicting, no? You make here everything, but very few of this is here. Also, this is the production place. But most of this ends up here. How can this happen? Maybe you can come up with ideas, but before, I give you a few minutes just to discuss with your neighbor, because sometimes it's helpful that maybe you have an idea, but you don't want immediately to come up with this, and you want to verify with your neighbor, could this be a cool idea? If not, you can also rearrange with the sitting. You three can also discuss a few minutes. Then we try together to come up with ideas. Is time still okay? How much time? Too much time. Okay, so I can see that some discussion goes on. That's very good. Is any team wants to come up with some ideas? You can sit and just tell me what you have discussed, even if you did not come to a solution, but maybe what ideas did you had in your idea, or what problem did you recognize? Someone wants to start from the teams? Here I think you were very intense, for instance. Someone wants to summarize, what did you discuss? Just come up, because this is to be honest, we don't have the answer. So don't worry. Cell biologists still study this. We don't have the full answer. So if you don't have an answer, it's fully okay. But we have ideas. And then the question is, can we maybe also test this experimentally, right? To see what of these ideas is correct or not correct. And this is what basically we are scientists for. We basically see you have data sets showing this lipid composition. You have data sets having this. Now you want to understand this. How can this be in the cell? Because on the plasma membrane, there is no lipid synthesis. There is no stereosynthesis. Stereosynthesis is here. So there's a dilemma for the cell. If this is the lipid composition of the plasma membrane, how does it get this type of lipid composition, right? Because normally, what would happen if you take here material and you would move it directly to here without anything else? You would get exactly the same lipid composition here, like here. What would be the result? Anyone wants to come up with just come up with discussion points, what you discussed? There was also I saw some people really discussing. What did you discuss? Okay. Yeah, so you are right. So you would basically make vesicles here. But this would imply that these vesicles now have a different lipid composition than this membrane. So you would need sorting. So you could ask yourself, okay, how could this budding process lead maybe to a sorting, to a specific sorting? Can you come up with ideas? Ah, okay, yes, you are right. There is basically, there's something wrong here with Thomas' drawing, because you have an in-between organelle, the Golgi. Okay, so you would bring the vesicles here, but then you would have budding here. But then at least the same problem occurs now on the level of the Golgi. So you take membrane from here, you form this membrane. It should be identical in the lipid composition. Now you make from this membrane, so new vesicles, bring it to the plasma membrane. At least here you need sorting now. Otherwise it will not work. No? That's true. But any idea how sorting could happen here by making a vesicle? Just ideas? Just come up. Whether it's, there's never wrong, or because there's many possibilities, but maybe only life has realized few of them. No? Because let's even make a larger turn. Amino acids. Why only we use 20 amino acids? Chemists know hundreds of amino acids, but life is only using 20 amino acids. Why this? Why would be coincidence? Yeah? Codons, okay. Oh, okay. What about L amino acids, D amino acids? Why is life based on L amino acids? Have you thought about that? I think there's so many fascinating questions, and we don't have necessarily the answer, we only have ideas. So the split, there's a type of asymmetry also in amino acids, L and D amino acids. And physicists believe that has something to do with the different interactions of the L and D versions with light. And in the cosmic, the light is very strong. So if molecules are exposed to the sunlight in the space, it's very intense. And if you interact differently, maybe some molecules are more resistant against the light compared to others, you get an asymmetry. So that's an idea that maybe D amino acids were more sensitive for light, and the L not, and they survived. And that's why we are. So there's different type of ideas. And when we come back here, there might be many ideas, but maybe not all of them are realized in the cell. So one way of sorting could be these proteins that would interact with membranes. So if I basically are a protein and bind, and I help budding, maybe I only interact with specific lipids. And that would basically be a type of sorting. So the vesicle that is here generated doesn't contain all the lipids, only a particular set of lipids due to this. That could be one mechanism, right? Now when we think about steroids, it's difficult because steroids, they have this head group. So very small head group. The rest is the steroid is, this is the hydroxyl group. Very difficult to see from the outside. No? You hardly see me. Phospholipid head group. I'm very, I see this. So, but what about steroids? They like to interact with lipids, for instance, that are more unsaturated. Because the unsaturated ones, what was the secret of the unsaturated ones? Do you want to come again or you want to come to me? One of you is enough. Okay, come to me. Should we go there? I think, maybe we don't need recording here, so it's more like for us here, like a tutorial. So do we want to be first saturated lipids? Yes. What would be your two tails there? Saturated fatty acids, no? Yeah, so here's saturated fatty acids. What do you know about saturated fatty acids? And I can do this, no? I can only not, I mean, they can do both this, but I can only stand on one leg. What is with you? Yeah, you need to be very flexible, you know. It's a carbon chain. And every carbon bonding can move. I mean, here, the legs. Yes, exactly. There we are, no? And of course, what we also can do, we can do this, no? Yes. Not necessarily with me. You can also go there. But once in a while, we might meet again, no? Yes. We can also do this sometimes, right? And then we can also do this, right? And we can do this. Okay. But since we are many, we would be very close. So, you want to be a steroid? You want to be a steroid? Yeah, steroid. You doesn't fit. You doesn't fit. No way to fit. Okay, now we have basically fatty acids that are unsaturated. And typically in biology, the position 1 of the glycerolipid, it's a saturated fatty acid. And on the second position is an unsaturated fatty acid. Can anyone explain what we two have now to do to look like an unsaturated fatty acid? A lipid with an unsaturated fatty acid. And what would be the consequence from the look? How can you see this? Because I don't want to open my leg. Yeah, and what else? Do you know this? Do you know the kink? Can you make a kink? Because you have now also. Yeah, like this. Yeah, this is okay. Yeah, stay there. So, and then you know, only your leg. Make a little bit more your leg to me. Yes. Leg stretch. Yeah, and you stay there. Yeah. Woo! A huge gap. A huge gap. Where's my sterol? Sterol? Wow! No, he fits. He likes us. We will go with him to the next membrane, you see? He will stay with us. So he will stay with the, unsaturated fatty acids because he will, just from geometry, we don't talk about even interaction yet about hydrogen bonding or whatever, which we could add on top of that. Yeah? But you see, and now, since he is basically associating, if you basically move us, you will move him along. Yes, sir. Yeah? So thank you very much. Perfect. Very good. So this is one way you can basically bring forward membranes with a different lipid composition. And now you go to the Golgi, and what happened in the Golgi? You make sphingolipids, glycosphingolipids. Actually, a part already happens in the endoplasmatic reticulum. And then there was this particular interaction of a sphingolipid with a sterol molecule. And that's why you get basically by moving forward membrane, but of particular lipid composition. You get the demixing. And this is what the cell has to do. The cell has to basically make sure that there is a sorting event. And the most sorting happens basically in the Golgi. Yeah? Okay, this I just wanted to give you, and sometimes it's just helpful, play this in your rooms this night, just a little bit to understand how lipids interact with each other and how these particular interactions can be actually exploited by the cell. Yeah? Now you maybe appreciate the complexity of a cell. There's a reason for. So this complexity is also needed not only to run these chemical basically reactions, but also to make the identity, to keep the identity of all these organelles. Thank you very much. Nice break. When do we want to continue, Abhishek, for the students? There should be some breaks, so I guess. 10 minutes? 10 minutes break? And then we are back and going into the secrets of membranes from the physical point of view again. Thanks a lot. Abhishek, you want to come with your own computer or you? Yeah, then we can shut down and clean. Yes. Very good. introduce. Yes, yes. No, but it's good. It's good. It's good when you introduce this, because it's also from the physical point, I think it's nice.
No, I just, people always say that, that how can, from whatever from the energy that I'm right now, what is the process of going to a shape equation? And
Super.
So then
Super.
you can take it.
Super.
And then from there you can take it
super.
over.
No, it's good. That fits perfect. Fits perfect. Thank you very much, sir. Cool. And you also have this big example. Oh my God, this example. And teaching. Oh, you are crazy. For me, it's of course good that we are all the able to manage this, because also for me now I have teaching free, so it's like easy for me to do good. Otherwise, teaching program is something also so for me it's not a stress as I would take that. Other ones, so the moment we are very lucky to do together. Yeah.
Very nice actually. For you, that's like pleasant weather.
Did you already have your big evaluation professorship, or does it come later? No, so yeah, you need to change your web page. You are still very bad.
My students have been telling that we run the
Because let's say when you are young, then it's okay. When you are getting a senior,
smaller? Okay. Sure. Oh, अच्छा अच्छा okay. हाँ ये रहा चौकी important. Thank you so much. Important. You can start. Okay. Good. So again, welcome back. I'm glad to see that despite the fact that this is quite a lot of theoretical stuff which is going on or going to be talked about today, you are still there, okay. And not disappeared. So let's see where we can reach, because this is the last lecture that I'll be giving, and there are a lot of things to cover, and obviously it is not going to be possible to talk about everything which regarding membranes. But what I'm going to talk to you about today is at least give you some flavor of what the physics, underlying physics is. I discussed yesterday about the morphology of the aggregates of these lipids and how they come about, and how there could be a simple-minded picture of this, which can give you a parameter which could be tuned to go from one kind of phase to another kind of phase, looking from spherical to cylindrical micelles to planar bilayers. Now, and towards the end of the lecture, I discussed about membranes, because now I'm going to look at it as a two-dimensional surface which is embedded in a three-dimensional space. That's what we had written down in at the end of the last class. And I also said that we have to, since we are talking about in terms of surfaces, we have to parameterize that. We have to, if you have to write down something for that, we have to do the parameterization of that surface, and the parameterization that we are going to look at is this Monge parameterization, and so this is called the Monge gauge. And the picture that we had was the following: that I have these x, y, and z, and I had a surface which maybe that's not enough. Difficult to draw it like that, but I hope that you can see that. And so this is the surface, and at every point it's like a height function that we are talking about, and so we give a functional form to that there is a sub f at every x and y. And also we discussed the fact that if you are having a system such as an overhang of the kind that we said that these are not kind of allowed in the parameterization that I'm talking about. So it's one parameterization, and obviously we can you can try to do better and try to see how things can be worked out. But anyway we are going to do some stuff which is going to be only scratching the surface. So and therefore we will just see how far we can go from just using this. So this is called the Monge gauge. And let's see where this takes us. So the first thing, of course, is that we want to describe the geometric properties of the surface, right? I mean, so what are the geometric properties of this surface? Now, if I look at this surface, if I look at a surface such as this, I will say, oh, this, one of the things would be the area of the surface, right? I mean, I take a local patch on that surface, and I want to find out what that patch is and what is the area of that patch. That is different, obviously, from a planar patch, right? I mean, I can take a projection of this take that patch and project it on that 2D plane. Then I would say, okay, that's like a very straightforward, that's a rectangular area element which will be given by dx times dy. If I have a small length along the x-axis and a small length along the y-axis, I would call that, on this, I would consider it as a patch which is of area which is going to be given by dx times dy. This is on the plane, okay, on this two-dimensional plane that I am talking about. That definitely is not the case when I'm looking at it at the surface. Okay? The area on that surface is definitely not going to be dx times dy. In fact, what it is going to have, and again this is something which you can show in this representation that I'm talking about, is that this, of course, first of all you can make a conclusion that okay, this area, once I take a dx dy from there and put it on the surface and I want to find out how that looks like, that definitely will be expanded. Okay, you look at it, it will be expanded, that area, small element of area that you have taken there, and that expansion, what is the form of that expansion? So my question is, what is that dA going to be on the surface? It cannot be dx dy because that was what was on the two-dimensional plane. What it is going to be is of this following form. And now this is, as I said, I'm not going to be proving these quantities. I'm going to make those statements, and what I want you to do is to keep that in your mind, and maybe sometime when you are comfortable doing the maths, you can work out how this comes about. So what you can write down is that this dA would be given by 1 plus, so I have a functional form like that. I can take a gradient of that. I'm hoping that everybody knows what a gradient is. Okay, so that's like the slope, right? It's like, when we talk about one-dimensional function, I will take the derivative of that function that will give you the slope. But here in this case, I'm looking at a two-dimensional function, so I'm going to write it in terms of the gradient. So like writing down this as grad of f, but the interesting thing is that it comes as a square of this. Okay? And so you can clearly see that this quantity definitely is bigger than the quantity that I defined as dx times dy. And so this is what I'm going to be characterizing the area of that patch on that surface. Okay? Now, which means that therefore I have to calculate the slope or calculate the gradient, okay, at any point in order to figure out what that area element change would be. Okay? So that's definitely one of the geometric properties. The area definitely will serve as one of the geometric properties which I have to specify for the membrane. Okay? But what is the most, kind of the most important property that stares at you when you're talking about a membrane? And the property that stares at you when you're talking about a membrane is the curvature. Okay? And you say, oh, this membrane is a curved membrane. It's curved this way. In this case, there is a curvature here, curved here on this other way, or it could be like that, or it could be like this. So this is definitely something that I need to worry about. So the second geometric idea, or the second geometric quantity that is very, very crucial when we are talking about membranes would be the curvature. So area is one, curvature is the other. And this, of course, is the quantity that we would like to figure out, and we would try to understand how we can get this. Okay? So how do we go about it? Now, so it turns out that I can do a simple, and what I'm really trying to figure out is not you're not talking about a curvature, you're talking about a local curvature, right? And locally, whether this membrane is curved at this point or is not curved in this way, in this direction at that point, or in this direction at that point. So what is the curvature locally? So if I'm going to do a local quantity, I'm going to find out, these kinds of aspects, then it turns out the best way to, the way to go about it, looking at the local neighborhood of a point on the surface, is to do a Taylor expansion. What do I mean by that? I look at a point at a surface and I say, what can I do by doing an expansion around that point? Okay, so of that function around a certain point on that surface. So that is exactly what we are going to do next, which is that if I have this function, then I know that if I want to Taylor expand this function about some point P, which has the coordinates, let's say x0, y0. Okay, so this is what I'm going to try to do, that I want to expand this in about that point which I have defined as, which I have given the coordinates as x0 and y0. Then this can be written down as f of x0, f evaluated x0, y0, plus, so I'm doing neighborhood of that point, so I'm just going around that point. And the first thing that comes about is, of course, now note that if you are familiar with one-dimensional Taylor expansion, then you would know that if I've taken a function which is like an f of x and I want to do the expansion about some point x0, then this goes about by saying that this is fx0 plus the next thing would be to take the derivative in this case, of course, since there's only one function, I don't need to give this partial sign. Anyway, it doesn't matter if you're not worried about it. It will just be like df dx evaluated at that point x naught. But now, of course, I'm saying that this is shifted from whatever x naught I'm talking about. And then the next one will be half of d2f dx2 evaluated at x naught again, x minus x naught squared, and so on. This is how I'm going to do this expansion. Okay? I'm hoping that at least some time somewhere. All of you have do come from a physics background? Yes, no, maybe. All of you? No, not everyone. Okay. So, but let's. It's difficult to proceed otherwise unless I assume this and talk about something. There will be some mathematical stuff that I have to talk about in order to come to the to tell you more about the membrane and how to describe the membrane in terms of the geometrical properties of it. So this is the kind of Taylor expansion that I would talk about if I'm talking about only f being a function of x. But now I have a function f being a function of x and y. So now instead of one derivative that I have here, I will have two to worry about. And in fact, what I'm going to do is to write this as sum over I equal to 1 to 2. And I will have a del f del. Now I am going to have to be careful because I'm going to ensure that this is like a, so I'll write this as an r naught evaluated at r naught vector. This r naught vector will signify this x naught y naught which I have talked about. So this is evaluated. This is the same thing that I'm doing here, except that now I'm doing two of them. And then I will have an x, okay, not x, but r minus r naught. Okay? Or ri, sorry. Ri minus ri naught. So let me just do that. Because there's a I which runs from 1 to 2. There are two of them, so I need to do this twice and get that correct. Okay. And then I would go to the next one and I would write this as sum over I comma j, which runs from 1 to 2. I will have a del 2f, and now in this case I will have a del ri del rj, and this again will be evaluated at r naught. And then I will have an ri minus ri naught times ri minus rj naught, and then I will have the higher order terms. Okay. Fine. Yes. Yes. Not Ri naught, but Ri. Yes, absolutely. Thank you. Yeah. Evaluated at R naught, yeah, which is given by x naught y naught. I take the derivative and then evaluate the derivative at the point of interest, which is x naught y naught, which is the point P. Okay. So this is the Taylor expansion. Now, what does this Taylor expansion tell you? Then immediately when you look at the Taylor expansion, you see that the first part of it, the second term actually, this one here, is basically again this gradient. It's a slope that we are talking about, which is this df dx. In this case, it's the gradient of f, which turns out to be important for evaluating the one geometrical property which I talked about already, which is the area. And maybe by doing this expansion, I'm trying to tell you that, oh, this second derivative which I'm looking at here will give me the curvature. Okay. I'm going to say that, but let's see whether there are more subtleties in this when I make this statement. Okay, so the first thing I do is to now, this one is already done. I'm just going to take that out of the board and tell you that I can, in fact, write down this in terms of a vector form. I mean, vectorially, rather than writing down x naught, y naught, which is in the terms of the components, I can write this as f, same thing, just rewritten as f of r. Okay, and this will be, of course, f at r0, because that's what x naught, y naught is. And then I have this one, I'm now going to write it down in terms of the gradient, because now gradient will have both del del x and del del y. That's what this quantity is. So I'll write this as a gradient of f, which is evaluated at r naught. And then I will write this as r vector minus, so dotted, r vector dot minus r dot vector. The second term, or the third term here, is going to be a little bit more tricky to write down, but bear with me. So what I'm going to do is to write this as r half of, it's a half factor, of course, it's important, keep it. And then I have a r minus r naught, the transpose, and then a quantity which I'm going to write down as h. I'll come to what that quantity is. And then I have the next one, which is r minus r naught, and so on. Okay. From what I have written down here, you can see that this is actually like a matrix multiplication that I'm talking about, because from what I have here, you can see that this quantity that I have has 4 elements to it. The combinations of I and j, I equal to 1, j equal to 1, I equal to 1, j equal to 2, I equal to 2, j equal to 1, and I equal to 2, j equal to 2. Okay? This will be the components of a 2 cross 2 matrix. And that's exactly what I have written down here. This quantity h, which I call as the Hessian, h is called the Hessian. If I write the Hessian down, it is just a 2 cross 2 matrix. The elements of these 2 cross 2 matrix are the following. So if I wrote down what this Hessian is, then this Hessian is the following. We'll have components del 2f, remember this is exactly what I'm doing, del 2f. Now when I equal to 1, j equal to 1, that is like both times it is actually a derivative with respect to x. So this is basically del 2f del x2. When I equal to 2, j equal to 2, then that's just del 2f del y2, and that will come here, which is the 1, 2 and 2, 1 components are the same. They are basically del 2f del x del y, or del y del x, it doesn't really matter. So I will write this as del 2f del x del y, and similarly here I will write this as del 2f del x del y. Okay. Right? Now this quantity, of course, presumably, and you will think, oh, maybe this is what, some of you may say that, oh, this is what curvature is. But I'll ask you to, hold your breath, because supposing I give you a very simple function. Let's say I give you a parabola, and I told you the parabola, of course, has an equation which is, since I'm writing down everything in terms of f of x, so I'll write down, let's say the equation of the parabola is actually f of x equal to x squared. Everybody will agree that this is a parabola. If you want to plot it, you will see that, oh, this is basically is going to, yeah, this is how this is going to look like. And you'll say, okay, now I can do an expansion. The expansion will give you, remember the first gradient is the slope, the double gradient will give you a d2f dx squared. Let's find out what they are. So if I do a f prime of x, that will give you a twice of x. If you do a double derivative with respect to, then this is going to give you this double primed is basically d2f dx. Okay, so maybe I'll just write it down so that there's no confusion. So df dx will be 2x, d2f dx2 will be equal to 2. Okay, so it doesn't matter where I am evaluating it. This quantity that I have is a constant. It doesn't matter at what point in the parabola I am going to evaluate it, right? I can do it at any point, it will still be the same number, which is equal to 2, which means it's a constant. Okay? d2f dx2 is a constant. So if you say that this is the curvature, if you claim that this is the curvature of that parabola, then I would immediately tell you, but this cannot be, because the curvature at the apex of the parabola is not the same as the curvature here, is not the same as the curvature there. Obviously, it is opening up like this. The curvature is definitely not the same, is equal to a constant everywhere on that parabola, evaluated at any point, but this is what you get. So how do we reconcile this? The reconciliation comes from the fact that it becomes the curvature provided that the gradient of f goes to 0. So this quantity which I called as a Hessian, which I have written down here, so this will define the curvature provided, okay, provided gradient of f is equal to 0 at that point. So this will give the curvature at a given point on the surface, provided that the gradient goes to 0. Can we apply it to this one-dimensional function and see? And the answer is yes. It becomes, where does the gradient go to 0? At x equal to 0. That's where the gradient goes to 0. And here, the curvature is actually like a circle. So if you think about it, it's like this is a curvature equal to constant is basically giving you a circle. The d2f dx2, which is a curvature, the radius of curvature will give you a half. So this is going to be a circle, which can be easily, you can see that this is what I will get at that point, at the point where I'm saying that this is at the point x equal to 0. There, if I take the gradient to be equal to 0, where the gradient becomes equal to 0, there this definition is definitely that of a curvature. Or other words, this quantity is definitely the curvature, and so is this going to give you the curvature, provided that this goes to 0 at that point. Okay. So now let's see whether. So that's a subtlety, and I hope that subtlety is in your mind always, and you remember that this is from a very simple functional form. You see that should be clear to you. Now, I have already given you the Hessian now, but I'm interested more on the surface, and I'm saying that, oh, this is what I have, and I'm also telling you that, oh, this is, I'm looking at a situation wherever I'm looking at a surface, whatever surface you give me, I'm going to only look at a region where the gradient is equal to 0. That point, a point where the gradient becomes equal to 0, and I know that if I calculate this quantity, then it will give me the curvature. So let's find out what that is going to give you. So I have this Hessian, which is of this 2 cross 2 matrix. Now if I have this 2 cross 2 matrix, the first thing that I note about this matrix is the following. Okay, so I hope I can take this off. And in fact, I can get rid of all of that stuff. It's anyway some bad looking expression. And I have this, okay, which I put out on the board. And the question therefore is, what can we say about this matrix? Now the first thing that about this matrix is the following, that it is a 2 cross 2 matrix, symmetric matrix, okay. And in such a case, okay, and this is again going to be news for at least some of you, this will be having 2 real eigenvalues. In other words, what I can do is to take this and write down the following equation. I can write down a matrix equation of the following kind. So I can have defined as, this is not something that I can go into details discussion about it, because I'm hoping that some of you, even if you remember the notes, and you can go back and find out what I'm talking about later. So what I can do is to write down what is called an eigenvalue equation, and write this as H, which acts on these eigenvectors. Okay, there are 2 of them. Then this will give you eigenvalues which are given by Ci, and then I will have the eigenvectors back. Okay? This is a special kind of a operation which, when acting on Vi, will give you Vi with some constant. Okay? And these quantities Vi, okay, are called eigenvectors, and the quantities Ci are called eigenvalues. What do they give in the context of the surface? Because that's what we are aiming for. In the context of the surface, what this gives, the eigenvectors will give you the principal directions. Okay? I'll come to an example, and maybe things are going to become a little bit more clearer. So this will give you the principal directions, and these eigenvalues are going to give you the principal curvatures. Okay? Let's take an example. Okay? At least see whether we can make some connect. I know that it's difficult. If you have not seen these, it will it will hit you hard. But let's see whether we can at least give an example and try to figure out how this can come about. So let's take an example of a surface. Okay. So let me just first write down the equation of the surface. F is a function of x and y. That's what we started off. That's the surface equation. And let's write this as half of x squared minus y squared. Okay. You put it in your favorite software which can plot a surface, and the surface is going to look like the following. A lot of you must have already seen this surface, or at least seen picture of this surface. It will be a hard one for me to draw on the board because I'm not very good at this. So that I can ensure that you see what it looks like. So this is basically and this is the back side of the surface. If you yeah, it's not bad. Yeah, I can yeah. So this gives you this part which I have shaded is basically the downside of that surface. And if I look at that surface, and I'm sure, and the question I would ask is, even if you did not know much about it, and I asked you, can you tell me how many how many curvatures are there in this? And you'd say, oh, there is one which is on that one, which curves up like that, and the other one which goes like this. Okay? If I go along the x-axis, so let's call this as x, that as y, and that as z, and I asked you that I will go along the x-axis, then you see that along the x-axis, the curve actually bends up, right? So let me just maybe draw it with some other color. A yellow one would have been nice, but let's see whether I have that. Yes, I have. So I will have, if I draw these two lines, and so this, of course, I don't know how to. This comes on the other side, right? I mean, so. But here on this surface, at least, you can see that that's how it will be. Okay. So this will come there, and the other one, and there are these two things that I have. And these are basically the curvatures that you're going to get. And the question is, does this analysis that I have just pointed out identify that? Okay? And the answer to that is yes. So what you can do is to first write down what the Hessian is. Now, you have to just quickly find out what this is. Okay? And write down that form. and tell me what these elements are. You have 1. Where minus 1? Diagonally. And on these ones? Will this give you x minus y? How many agree that it is 0? Only few hands. So not everybody is convinced it is 0. It's 0? 0, okay. Yes, it's 0. Okay. Actually, a lot of you, some of you, if you know this, then you will immediately say, wow, this is good because it's already diagonalized. This Hessian that I have written down, this matrix that I have written down is already diagonalized, which is brilliant because that means that they are already the corresponding, these directions are the principal directions. So they are, I don't have to do anything, any more massaging with this, do transformations and all that stuff to get it diagonalized. So this is already diagonal. And therefore the eigenvalues that I'm going to, and I can correspondingly find out the eigenvectors, and I want to find out, and I can ask you this, do this exercise, go back and do the exercise and find out what the eigenvectors are. Then V1 will be 1, 0, and V2 will be 0, 1. And what are the eigenvalues? 1 and minus 1. Thank you. C1 is plus 1, C2 is equal to minus 1. The plus 1 is along the, as I said, along the x-axis. Okay. Now, there will be the curvatures. There will be the principal curvature, there will be the curvatures provided that the gradient is equal to 0. The gradient of the function is equal to 0. Where is the gradient of the function goes to 0? When? X equal to? No. Gradient will go to 0. What is the condition that in this case the gradient of this quantity is going to go to 0? X equal to y? Can I claim that? Okay. So if I were to have that line, and on that line, whatever that x equal to y line is, if that's what you are saying is true, okay, or whatever you find out, if along those points, if I evaluate this quantity, then this is going to give me the curvatures at those points. Provided, as I said, I am always saying that wherever gradient of that function is 0, if I want to find out what the curvature of that surface is, that is going to give you the curvature. I'll evaluate this quantity, whatever this Hessian that I have evaluated, and therefore this C1 and C2 that I have found out are therefore the curvatures. And I can say for surety that these are what they are. Okay. Now you'll say, okay, this looks very nice, and I'm happy with this, and therefore my job is done. But my question would be that eigenvalues and eigenvectors are not the best way to look at it, and why not? The point is that I can take that surface and do a 90-degree rotation. Now, nothing happens to that surface, but will I change the eigenvectors and the eigenvalues? Will they remain invariant? The surface remains invariant because, okay, it's a surface, and I do a rotation. I mean, the surface is still that same surface, it's not changed. The character of it, it's still two curvatures, which is C1 equal to plus 1 and C2 equal to minus. Okay. But, okay, so we have to figure out. I'm not saying maybe it has, if I do that 90 degree, C1 becomes equal to minus 1 and C2 becomes equal to plus 1 for you to check. But if I do that and I say that, oh, this is going to change, then it means that I'm talking about a different set of eigenvectors. The eigenvectors will swap, and so will the eigenvalues. And if they have swapped, then, they are not invariants, right? I mean, doing something and then it doesn't suit me. Such transformations is not going to keep those things in. Are there some quantities which are going to be invariant irrespective of what I do, these kinds of rotation that I showed you, the example of a rotation, in the context of the problem that I'm talking about, the Hessian, the matrix, the 2×2 matrix that I have written down? Very good. So, absolutely. So for this you don't, so as I said, these are very, simple things which are there, as long as you have, it's go back a little bit of eigenvalue equations and all that stuff, matrices. As long as you've done that, you will see immediately that these are the quantities that will stick out, clearly come out. And so the two invariants that we are going to look at, okay, are the two invariants are therefore the sum of the curvatures, okay, the what is it called, the trace and the determinant, and therefore the definition is that the trace would just be C1 plus C2, and this I'm going to call this as J. Okay, and the determinant is clear. At least the example, if you just keep in mind this example, then also it will work. The determinant is basically C1 times C2, and I call it K. Okay? And this first thing is what is called the total curvature. Sometimes this is called the total curvature. Sometimes you will see in textbooks of membrane biophysics, you will see the thing called mean curvature is basically half of taking the C1 plus C2. Whether you take it C1 plus C2 or you take a C1 plus C2 divided by 2, it really doesn't matter in the grand scheme of things. So I'll just keep it as total curvature. The other one, which is the product of these two, has also a very good name, which is called the Gaussian curvature. Okay. So what have I done so far? What I have done is the following, that I've taken a surface, I went into some parameterization of that surface, said, okay, this is what the surface is, how the definition goes about. There I wanted to find out, okay, what are the geometrical properties of that surface? In order to get the geometrical properties, I note that both the gradient and the curvature are important geometric quantities, and that comes about in the Taylor expansion about a certain point locally. If I do the Taylor expansion, that will give me both of it, and both of them are important because the gradient will be used in calculating the change of area, because remember that's where the gradient enters the picture. And also there's a curvature part which I have written down, and I said that, oh, that's a second order, but it is not necessarily a curvature. It becomes a curvature wherever at whichever point the gradient of that function goes to 0. Wherever at any point on that surface, if the gradient goes to 0, I evaluate the second quantity, which is a Hessian, and then that will give me the curvature. Okay, this we established, and then we said, oh, the Hessian, it's a 2 cross 2 matrix that has real eigenvalues, that is all fine, and this is going to give me the curvature, but not necessarily, because there is a problem, because now I can get hundreds of possible principal. Every time you do some transformation, I will say, oh, there is another direction and another. That's not a good way, because eigenvectors and eigenvalues will change, and they will say, oh, that's another direction. Then there's another. So I can come up with infinite such possibilities, but that's not a good way of looking at it. So I said, so then what is a better way of doing it? I said, oh, let's look at the invariants, because I know this, that the invariants are the trace and the determinant, and the trace and the determinant are going to give me two quantities which I'm going to define as the total curvature and the Gaussian curvature. And this kinds of gives me a very nice first-hand understanding of what this is. Of course, the bigger question is, what do we do with it? Okay? Yes, okay, you've done some analysis and you found out this. What is the next step? What does this have to do anything with what Thomas works with, right? I mean, what has to do with a vesicle or a lipid, lipid bilayers and all that stuff? Okay, this is where we are going to get to. So now, where does this come about? Is to say that, okay, I look at the lipid bilayer membrane, And the lipid bilayer membrane that I have, I know that it's a fluid membrane. Okay? Now, although it's a fluid membrane, and you say that, oh, fluids do not support shear and all that stuff, I mean, they will just, if you shear a liquid, it will just flow, right? I mean, there's no point in. So, of course, when you talk about fluids, for those who know the language of fluids, you know that it's about the rate of shear. These are the quantities of the strain rate. That's what becomes most important. But I'm not going into so much details. All that I'm going to say is that any deformations, even in the context of a fluid membrane, if you want to deform the membrane by whatsoever way, I told you before that it is like, the extent, the physical extent of this membrane with the thickness and the lateral extent that looks like a paper or something like that. And the question is, what about trying to stretch it? Will that cost any energy? What if I want to bend it? Will that cost any energy? Okay. Now I'm trying to ask these questions, and then therefore I'm going to make this connect. And the moment, you can imagine that the moment I'm going to talk about stretching, that will involve changing the area of the membrane. The moment I'm going to talk about bending, it will involve the curvature of the membrane locally or wherever it is. So now you can connect to what I was talking to before, that why these geometrical quantities will play a role when I'm going to talk about the membranes or try to create a physical picture of what these membranes would be. That is where the role is going to be. Okay. So how am I going to do it? So let's try. The first thing is to say, okay, let me maybe find out what stretching would cost me if there is an energy that I have to write down. So let's try to do that. So I want to stretch it and consider therefore membrane So it's like a 2D sheet, and relaxed state, it has some area which I call as A naught. Okay, that's the area when there is it's just no problems with it. I'm just taking that, and now what I want to do is to say that let me just try to stretch it so that the area changes, the area increases to some A which is greater than A naught. That's what I'm trying to do. Now, what is the immediate quantity that you want? Again, I want to always think about things in dimensionless units and all that stuff. So dimensionless quantities you talk about when you talk about stretching a line or something like that. Then you say, oh, change in length to the original length. Okay, you always talk about things with respect to the original length. That's what will give you the relative change. That's a relative change. So in this case, if I want to say, what is the relative change in area that has happened? I will say, oh, the relative change is going to be A minus A naught, that is the delta A that I have done, divided by A naught. That is what I was going to call as the strain. Okay, so the area strain. So this will give me the area strain. Okay, and let me call this as small s. And now I want to write down the following quantity. I want to write down a stretching energy density. Okay, I want to just phenomenologically write down what the stretching energy density will be. It will cost me some energy, right? I mean, I can immediately see that there is if I'm trying to do that, I try that with a paper, it is not going to just, you don't stretch it and it will tear off. Okay? In this case of paper, if you do too much of stretching, it will tear off. So stretching, I'm going to calculate stretching energy density. Energy density means energy per unit area. And I call that some E stretch. And I want to write down what this is going to be. Now, remember I'm trying to build up a theory here. I'm saying that, okay, what am I going to do with this? I know a quantity which is important, which is the strain. The strain is the most important quantity. Now, will this energy density be linear orders in strain? Should it be quadratic orders in strain? I mean, I could have write down something as something s plus something s squared plus something s cubed. It could be different functions, right? I mean, I'm just phenomenologically trying to build it up. I'm saying, okay, this could be, something s plus something s squared plus something s cubed and so on, right? I mean, so I don't know what that is, but I'm just trying to figure out. And the question is, okay, so will this work? Can I keep it s? I can't. I can't because I've already told you that the minimum area is A naught. So if I want to take the derivative, it cannot be a linear function of s. It just cannot be, because that will not give me a given A naught. I've already told you the relaxed state is A naught, and I'm changing with respect to the relaxed state. So if I think about trying to do the derivative and then putting it equal to 0 and all that stuff, then obviously I cannot have the first term to stay. Okay? So this one cannot be there. I cannot have a linear term. And therefore, if I ignore that there is a strain, but that S is a small quantity, right? It's delta A by A naught. Delta A, if you do not make a large change, right? I mean, you only want to make small changes. And so delta is small with respect to A naught. So that number is anyway very small. And so S squared is already small. S cubed will be further small. I don't want to go into that. So up to some orders, I'll say, okay, so this is nearly equal to this quantity. Well, I'll give some, some form of this quantity which I have drawn, which is half of, why I bring that half, it's not really important, but you just, this is what it is, okay? So, don't ask me why I bring this half, and just some weird person just writing a half factor, which I could have absorbed it anyway in that Ka, but keep it anyway. Okay. So what is this? Yeah, this sounds convincing, right? I mean, this is what I would be able to do. Which means that I'm going to therefore write this as half of Ka, A minus A naught divided by A naught whole square. That's what this comes about. Okay. So that's the stretching energy density. But I'm really interested in the stretching energy, not the energy density, but energy for the entire membrane. Now the entire membrane has the area, relaxed area as A naught. The relaxed area of the membrane was A naught. So if I want to find out what that total stretching energy is, then I would get the following form. So the stretching energy would be just taking this and multiplying with A naught. It is just taking the E stretch and multiplying it with A naught, and therefore I will get half Ka, and this will be A minus A naught whole squared divided by an A naught. Okay. And pleasantly enough, this experiment has been done to death, trying to stretch a membrane. Okay. Different ways of doing it. Okay. And I urge you to look for how you go about doing it and measuring this Ka. Okay. This will be the stretching modulus. What is the stretching modulus will give you? The stretching modulus Ka is of the order of 240 millinewtons per meter. You can try to find out what the energy scale is, divide and the fact that, find out what the unit should be. Yeah. Now, you can do a small experiment with a lipid vesicle. Yeah, take a lipid vesicle and say, okay, I'll take a lipid vesicle and maybe try to strain it, and this could be a homework exercise or a tutorial exercise which you can work out. So let's say that I have to use my specs because I'm really poor in. Yeah, so I have a let's say I have a vesicle which has a radius of r. Okay? So this is like a tutorial exercise. I'll not be there, but I will ask whether you have done it or not. Find it out. So tutorial exercise is the following, that I have a vesicle of radius r, sphere, and I want to increase this. Okay? So I'm going to increase this by a fraction. So the increase is by a fraction p. Okay? I'm increasing this as so r goes from r to r plus rp. Okay? Or r becomes capital R times 1 plus p. That's a fraction. P is a fraction. Okay? Now, first find out what the strain would be. Okay? Then find out what the delta E stretch, the change of this energy that you are going to find out. And this you find out for a vesicle for which the change is, or the fractional change, so you find out what this quantity is first, the form of this quantity, and then put some values into it, numbers, because those are interesting, right? Because it's just not mere numbers just for there's a meaning to it, because they are doing experiments now. You have experiments which says what that Ka is, and this is a value that I can take and put it there, and say, okay, maybe the R, the vesicle size, what would be a typical vesicle size? What would be the radius? 10 microns, okay, fair enough. Okay, that's a radius. So, okay, and the fractional change is, let's say, 0.1. Okay, making a only small 0.1 change. So P is equal to 0.1. And find out how much energy it would cost to make that change. Any guesses how much could that be? In terms of kT, kT is a unit of energy, right? You talk about thermal energy, kT. So, what orders do you think you would get? 10 kT, 100 kT, thousands of kT. Okay? So find it out. And you will find that stretching is a very costly exercise. It's a fluid membrane. Yeah, you call it a fluid membrane. All the time you talk about it as a fluid membrane and all that. Stretching turns out to be a very, very costly exercise. Okay? This is where you put in numbers. This is where the theory makes connect with what we are talking about. It's not just some figment of my imagination, but something which is there and which you can actually do and find out. Okay? So that's the first thing. And now I have, I'm already very, very bad with the time. But let's see whether I can get to at least one part of it, the final part which I want to get to. So you see that area. From there I got the stretching energy that correctly done, everything was fine. But now I have to also ask this question, that what is a bending energy that I can work out. Okay? So what is a bending? So membrane stretching is one. What about membrane bending? You see, once I have done the previous exercise, this stuff is actually pretty straightforward. And what do I say? Why do I say that? Because I'm just trying to build up a free energy kind of a argument from saying that, okay, what are the minimal symmetry allowed terms, and what are the energy allowed terms which are going to be there, and therefore I'm building it. So membrane bending. And I ask the same question, that what would be the bending energy density in this case? Okay, and I will tell you, okay, I ignored a constant there in the stretching energy, but I would have kept a constant, it would not. But supposing I just had this here, and this is important here, at least this context is meaning to that. Okay, so I will say again something plus the first order term, because now I am going to talk about bending in terms of the curvature. Just like I talked about stretching in terms of the area, bending would imply that I change the curvature locally, and that will cost me energy. And therefore, what are the quantities that I have as invariants which I have defined for which I have curvature? One was the C1 plus C2, which is the total curvature. The other was the C1 times C2, which was the Gaussian curvature. So those are the quantities that I know, J and K. So I have to, whatever things I'm going to do, this energy bending, the energy bending energy density should be in terms of those. Should be some function of J, should be some function of K, could be J, J squared, J cubed, or K, K squared, K cubed, whatever. So let me just try to again write down the same way as I did it for the strain when I wrote it for the energy, for the stretching energy. What would this be? I'll say, okay, the first thing that will come to me is the linear term. The linear was the C1 plus C2 is a J. J is a linear term, right? I mean, there's no, the curvature, linear dependence in the curvature. So I'll say, okay, that would be J. That's the first. K cannot be, K is already a quadratic. K is C1 times C2, so it's a quadratic order, second order. So I can't keep that now. But can I create a quadratic in J? I say, yes, I'll just square it, J squared. That can easily come in the next stage. So I'll say J squared. These are the coefficients which I don't know anything about. Okay. Then I'll say, okay, this is not the only quadratic term that can exist. I can also have K. So I will have, and then dot dot dot, because then they are the third order terms and I'm cubic and whatever, and I'm not interested. Just like as I did, I kept only up to terms which are higher orders. I'll just neglect and say okay. Looking at small curves, curvatures, costing, not humongous ones. You don't take a, well, you can take a vesicle and try to put something inside it, which you obviously do in a cell. That's another exercise. I do not want to get into those stuff, but let's see whether we can build the most simple kind of thing stuff here. It'll work like a, it really does work. I mean, so it's quite nice. Fine, this much is fine. So might tell me now what I'm going to do with this. So, well, I say that, okay, this looks like all that I can do. Maybe there's a better way of writing this. And the better way of writing this is the following. The better way of writing this is, is to identify what this term is. And this term here could be a number of things. It could be the surface tension. Coming out as a sigma. Okay. This could also be chemical potential of lipids. I would rather keep the surface tension and write this in the following way. So I write down E bend as sigma plus. Now I can take these two terms here and combine them to form one term. I take the J dependence and the J square dependence and combine them to form one term by writing it in this way. I write it as a half and kappa and write this as J minus J0 squared. Okay. What is that J0? I'll come in a minute. Wait. Okay. I'm building a phenomenological theory anyway. It's okay. I'm just allowed to do this because that's like I'll have a J squared, I'll have a J0 squared, whatever that means. That could be incorporated in the constant, whatever is there. And then I have a J0. That's also fine because that J0 is whatever is a fixed quantity. So it's like J minus J0 whole squared, and therefore it's fine. And then I have plus kappa bar K. This, of course, is the bare surface tension. Kappa is a bending modulus, all very important quantities. J0 is the spontaneous bilayer curvature or the spontaneous curvature. And finally, this kappa bar is called the Gaussian curvature modulus. So if I have a membrane which has an up-down symmetry, this doesn't have an up-down symmetry because what I have written here is not the same as what I have written there. But I could have taken a white page, which I, yes, I have one. Good. So without notes, I didn't get time to reach there. So this is this one and this one. There's an up-down symmetry. If there is up-down symmetry, I would claim that I cannot have a linear term, J term, because J was C1 plus C2. If I have an up-down symmetry as opposed to if I do not have an up-down symmetry, this is what I'm trying to claim, that in the presence of an up-down symmetry, this should not matter, because if I do that, if C1 plus C2 will change, C1 plus C2 is going to change if I have an up-down, if I don't have an up-down symmetry. But if I have an up-down symmetry, it doesn't, so therefore I will not have this term. This essentially implies that I will not have that J0. That's a spontaneous curvature. J0 equal to 0 is where I have an up-down symmetry. Okay? So if I keep this and now write down the corresponding equation or the corresponding energy density, not the energy per unit area which I have written down here, note that unlike this surface where I just took the stretching energy density and multiplied with an A0, I cannot do this here because I am locally there are lots of quantities which are going to change. So what I have to do if I go to from energy density to energy to the total energy, I have to integrate over the total area of that surface, whatever surface I have. So that is what I'm going to do now and write down, not do the problem, of course, there is no way I can go beyond that. I'm going to say what is E bend, and this is famous. This is actually a functional, anyway, I will just keep it that way and not, this will be dA, and then I have sigma plus half kappa j minus J0 squared plus kappa bar k, and this is called the Helfrich, famous Helfrich energy or Helfrich functional, energy function. So what do you do when you are trying to find out, which I cannot do of course at this moment because I'm already quite late, is that if you want to find out vesicle shape equations or different kinds of shapes that you form, is do a minimization of this energy functional. You do a variational minimization of this. So it's essentially, if you know about it, it's Euler-Lagrange formulation. You do an Euler-Lagrange formulation, you do a variational just as you do for any surfaces that you have, classical mechanics would have taught you. So you do a variational with respect to this and find out what this is going to look like and put it equal to 0. So minimization is basically extremization is equal to put it equal to 0 and then find out what that is going to give you. That is going to give you what are called the shape equations. Okay? So that's what is going to give you the shape equation. So this one is, although I started off with the Monge gauge and all that stuff, this is not specifically this thing for the Monge gauge because that is not, I've not written down in terms of the dx dy and corresponding one which I can do in the Monge gauge, and I find out what that is going to give me. So I can do that and find out what that shape equation is. Okay, that is number one. Number two, and that's also very important. That's what Tripta does a lot of times, is to look at vesicle fluctuations. Look at a vesicle in that, and I look at thermal fluctuations of this. And the natural question is, how can I find that out? Where from will I work that out? And again, that will involve doing a Fourier transform of this energy that I have written down there. Okay? Or in fact, if I write down the corresponding thing in the Monge gauge and then do a Fourier transform, what you will find that form is going to give you this. So I'll just write down this and then stop. I hope that I'm allowed that time. Is that the E bend in that case would be given by, when I do a discrete Fourier transform, you will get it as a half L squared sum over the Q modes. This is something for you to show for people who are interested, can go into more details and write this as a sigma Q squared plus kappa Q to the power of 4. Okay? And now if you identified this, basically for people who know harmonic oscillators and all that stuff, it's very nicely separated out because it's just the sum of each one of them. They are not, there's no interlinking between the two. They can sum of these harmonic oscillators. So each one of them will have a half kT. Then I can define what is f mod of f tilde q squared, ensemble average of that, and this will give you a kBt divided by L squared into sigma q squared plus kappa q to the power 4. And if you ask Tripta and look at vesicles or ask Thomas and find out what this, so you essentially find out what these coefficients are. Q squared will be when you have a sigma, when there's a surface tension. And you can fit the physical fluctuations with this and determine what the sigma and the kappa will be for this system. You see? This is how you fit the entire thing and find out what the sigma and the kappa for that particular system would be. So if you put some proteins on the surface of this membrane and the membrane's rigidity changes, then you will fit again and find out what the kappa is now, what kappa has happened, because you know what the modes are, because you can do that with respect to the modes. And so this is what is called the mode analysis. But anyway, I don't want to get into the details about it. Just wanted to say that this is all. So this stems from this one, and from here you can get the shape equations. You can also talk about energy, about the fluctuations of the shapes, and up to some level this works like a charm. Okay, I will stop, and unfortunately I don't have time to go into details. One quick issue that somebody may point out is that somehow in this form that I have written down, this has completely gone out of the picture. Where is the kappa bar? Okay, I wish I could answer that. Well, I know the answer, I'll tell you, but just don't ask me beyond it. There's some theorem in differential geometry called Gauss-Bonnet theorem. Now, Gauss-Bonnet theorem turns out that if I apply that theorem, put in some boundary conditions. So what happens is this term contributes only when there is a topological change or there is a boundary that is there. Only then this is going to keep come into the picture. If not, then this just comes as a constant. So in most cases, in a large number of situations, you will find that the Helfrich energy is just written down as half kappa of the curvature squared. Okay? It's just written down as a half kappa into the curvature squared. In fact, if I forget about a surface and I just had a line, which is what a large number of polymers, which are called semi-flexible polymers like microtubules or mortar proteins and all that stuff are all polymers, if they want to be described and their bending also, I can think about a bending of that line, then also you will get us exactly, we also write down a Helfrich Hamiltonian of the kind of half kappa, and then I will have a del 2r del s2 whole squared and then ds. So that's how I'm going to do it. Exactly what I have written down, except that now instead of the surface, I have a line and I'm doing a derivative, a double derivative of this, which was of course what this is. J is, remember, the curvature which has double derivative, and the square of it, so that I do not, this is not, this and that are equally penalized. You don't penalize this one more than this one, and that's basically the whole square takes into account this factor. Okay? So what Helfrich Hamiltonian or the form of it actually applies to a large context of soft matter or several places. And so I wanted to give you a flavor and tell you that this actually is something which can be worked out. The details, of course, I have not been able to completely show you, but you're free to look at literature to find it out. Okay, thank you.
Any questions?
Yes, I'm happy to take them, but they are already like.
I mean, he's around, we can also talk. So it's a break for lunch now. So I request the participants go together to exits. So some of the people from my lab